Hi everybody, it is 12:30, so that means it is time for our Translocation Symposium Day 2. We've got a really engaging session coming up. If you attended last week, I'd like to say thank you for participating. The recording of that is already on the ANPC YouTube channel, so you can have a look if you missed anything. And we'll be sending out feedback forms for any anonymous feedback later today. Now, this session I'm particularly excited about because there's some quite obscure titles involved. We're talking fungi, orchids, mistletoe, field monitoring, and working with the right plant in the right way. The symposium has been prepared by the Australian Network for Plant Conservation. It's been funded by the Ross Trust, making it free for everybody today. My name is Chantelle Doyle. I'm going to be guiding you through the sessions. I'm the project manager for translocation at the ANPC, and I'm also doing a PhD on plant translocations. Now, a little bit of housekeeping. As with last week, we won't be pausing for questions, but all the speakers are happy for you to pop a question in the Q&A. We think we've got that problem sorted out, but I'll let you know. If you have any problems at all, you can put those in the chat section, and Joe and Christine from ANPC are running around busily in the background, making sure all of this runs smoothly. Now, a little bit different to last week, I'm going to be introducing each of the speakers just before their presentation to give them time to get their slides in order. And then once I finish introducing them, they'll be ready to take it away. Now, to start, I would like to acknowledge that I am meeting today from the lands of the Gadigal people, and I would like to acknowledge traditional custodians of the various lands on which you are joining from today, and pay my respect to elders past, present, and emerging. I would also like to extend that respect to any other Aboriginal people joining us today. I would invite you to please acknowledge the country that you are joining from in the chat. And while I do that, I will ask if Amos Atkinson is here yet. Is Amos around?
No, I can't see him, Sean.
No, okay. So Amos is going to be speaking this afternoon about the Right Plant, Right Way project, and he will also be speaking about his country as well. Okay, now we are ready to get underway, and you may have noticed that we don't have any orchid experts joining us in person in these sessions, and that is because, unbeknownst to me, July is absolute peak orchid planting season, so not good timing for them. But we're really fortunate to have a personal on-site orchid translocation training session that has been submitted from the Wild Orchids project. The project has been run by Murray Local Land Services, but it's partnering with the New South Wales Saving Our Species program, RBGV Victoria, and the Australian Native Orchid Society. Now, if you've ever wondered why it takes so long to plant an orchid, this little field video compiled a couple of days ago might answer it for you. Enjoy.
We basically take the plants out of the pot, very gentle. We separate them, and we take the measures from the old tuber. So we take the measures from the plant, from the old tuber. This is the new tuber. And here's where the mycorrhizal fungi is. So it's really important that we don't damage that part. And we take the measure from the leaves, the old tuber, and we keep in the tray. Put in a tray with paper towel, so we keep everything wet and protected from the sun. It has a tag, each plant, so we know the exactly location, and they're ready to go.
Okay, so this is the Oatlands Dwyer, and what we've got here is the old tuber, last year's tuber, these fawny coloured ones. We've got the new tuber that's forming this year, the white crystalline-like tuber there. There's a taproot down the bottom, which we need to be careful of when we plant, and these little guys here are water roots that sit out level with the top of the tuber and absorb moisture for the plant. The ground level on the plant is just here where it's changing from white to green. So when we're planting, reintroducing these, we need to dig a hole deep enough so we don't crush the taproot, and then position these water roots correctly as we fill the hole back up to keep the soil level here where it's been growing in the pot. We then line the plant up with the edge of the ground and hold it against the side of the hole. Then lift the water roots out the way at this stage and have them supported. Then grab the soil and crush that up finely, slowly filling the hole as we go. And then slowly tapping it down, but not crushing anything. And making sure the water roots are still up at this stage.
We sort of team up and do it.
And then once we get up to the level of the water roots, we leave them, get the little Romulia out the way, we put them back down onto the surface of that, and then continue to fill. Lightly packing as we go. And there we have it, another plant in the ground. For future monitoring that we do is put a tag in the ground 100 mil to the north of the plant, so when people come back next year and in years in the future, 10 years, whatever, to monitor, they can locate the tag and find where the plant is.
Well, I've never done any orchid planting, and I'm sort of glad I hadn't. I'd been warned that it was quite laborious, and now I understand why. Really interesting though, and that one was filmed just a couple of days ago from the field. So sorry about the sound there. Next up, we have Leonie Monks. Leonie, if you're ready, you can get on the screen. Now, Leonie is from the West Australian Department of Biodiversity, Conservation and Attractions. She's undertaken dozens and dozens and dozens of plant translocations in Western Australia, as well as authoring numerous publications and guidelines to share her learnings. And in this talk, she's going to be focusing on what she's learnt from the field in terms of implementing, maintaining and monitoring translocations. Take it away, Leonie.
So yes, I'm Leonie Monks. I'm a research scientist with the WA Department of Biodiversity, Conservation and Attractions. And my research focus is purely on plant translocations, which I've been doing for quite some time now. So I've been asked to use my experience in this area for my talk today about implementing, maintaining and monitoring plant translocations. And so what I did was I based my talk on chapters 6 to 8 of the translocation guidelines. So I'm sort of going to talk through some of the guidelines and use my experience to highlight these guideline points with some case studies from the field. Let's just use that. Oh, there we go. Okay. So generally following the preparation of a translocation plan is the collection of propagation material and propagation of plants. And we've had some great talks last week from both Megan and Warren giving some good overviews of this process. And so I won't go into that in any depth except to highlight the value of having really good relationships with people who are collecting the propagation material and propagating the plants for you. I'm assuming that's not you. But having a good relationship will really allow you to work together to overcome any issues and get the timing right for propagation and ready for planting and working together to get the best possible outcomes. So we found that's really valuable just to make sure that we continue to maintain that really good relationship with our propagators. I'd also like to really reiterate the point made, I think, by Warren about the importance of labelling the propagation materials through the collection and propagation process, so you know exactly what you're planting. And the example I've got up on the screen here is of 2 subspecies of Hibbertia echinata, both of which we have translocation programs for in WA. And what we found is the labelling was incredibly important for these ones because we had a mistake made when we were picking up plants from the nursery, which meant we accidentally collected several seedlings from the pink flowering Hibbertia echinata, which is on the left there. And that grows over near Esperance. And we accidentally planted them at the site of the yellow flowering form, which is Hibbertia echinata occidentalis. And that occurs several hundreds of kilometres away to the west near Busselton. It was only when we were looking at our nursery accession numbers, that when we were checking them off against our list, that we realised our mistake and we had to actually pull the plants out of the site. But without adequate labelling, we might not have realised this mistake until the plants flowered several years later. And it might have actually been too late to prevent hybridisation. So adequate labelling and double checking the labellings is incredibly important. The next point in the guidelines is about logistics assessment, and it's making sure you have everything ready to go for the translocation ahead of planting. So things like the site infrastructure. So in the photo here, I've got a boot cleaning station that's installed prior to when we're doing the translocation. So we're ready to go with our hygiene before we start the planting. And having all our equipment and things like that ready. So we've got in the back of the car, there all our equipment loaded up in the bottom of the car, like shovels and drills and things like that, and our irrigation poly pipe tied onto the roof, and we've got our plants all packed into the car there ready to go. So just making sure you've got all that ready to go so that you're not scrambling around on the day trying to get equipment. Also ensuring that the site's ready to go. So for example, if you have to get all the weed control done and things like that, that's all done ahead of schedule and well in time for the translocation to happen when you need it to happen. And getting all the people lined up ready to help you too. So the photo I've got down the bottom there is where we did planting of Verticordia spicata. They're all holding all the plants we had ready for that site. So we had plenty of people for that translocation, and we got it done in record time. But we did actually have other sites to plant with this species, so we managed to keep everyone busy for several days. The timing of translocation will depend on several factors. So in his talk last Thursday, Warren covered getting the plants ready, but other factors will play a role, such as making sure you got the site infrastructure done ahead of time to avoid damaging the seedlings. So we've got down the bottom there some of our guys getting the tank stand ready to go, and we made sure that that was done ahead of time because they were lugging around, pine logs and trampling through the site. So we made sure that that was done well ahead of when we had to get delicate seedlings in. Also thinking about when the conditions at the site are suitable. For us, it means getting the planting done in winter, but this is obviously going to vary according to the different climatic zones that you're looking at doing planting. and other sites such as other factors such as whether you can access the site. So the top photo here is of our track that got washed out before we could actually get to the site to do the planting. So those sorts of things need to be taken into consideration. So for us also, how we access the site in wet soil conditions is particularly important because Phytophthora is a big issue for us. So we might need to alter how we access the site to avoid patches where we're worried about that Phytophthora might be, or going through large sort of muddy puddles and things like that. So those sorts of things you need to take into consideration as well. And also for us, and it might be the case for a lot of other people, funding is another factor that influences when we do planting. So we often need to acquit accounts before end of financial year, which means we're planting sort of at the start of May to ensure that that's done ahead of time. And then finally, the issue is of planting, whether you plant in 1 year or over multiple years. So we generally plant over multiple years for logistical reasons. It's much easier to do smaller plantings over several years because you make sure you get it done well and you can really look after the plants. And we want to also hedge our bets against poor environmental conditions. So maybe you'll jag a good year, and one bad year, so that at least some of the plantings are successful. But if you have the time and the careful planning to do one large planting, it may be a better option for you as well. So the other thing that talked about in the guidelines is considering designing these translocations as experiments. And there's lots of papers and articles in the literature that talk about the value of using experiments to learn from these translocations. And we can do these as either small-scale experimental translocations or what we call pilot studies ahead of the main planting, or they can be undertaken as part of the main planting. So examples here, we set the translocation for acacia apica up as a small experiment where we looked at the value of using mulch to help improve survival and growth. And the results here are just for survival, but they showed little difference in survival over the short, medium and longer term between plants given mulch and those not given mulch. And the growth also showed the same pattern. So we found from this small scale study that in this case, for this species, the mulch was not effective and we didn't use in subsequent translocations or subsequent plantings of this species. But as an example of a translocation where we, an experiment where we implemented it as part of a full translocation, this is Banksia cuneata, also known as the matchstick Banksia. This is a serotinous species and predominantly recruits after fire. So we knew that fire was an important factor in the species life cycle. So we're interested in assessing whether burning the site prior to planting would assist in the translocation establishment. So we burnt a patch of vegetation at the translocation site and we left another patch unburnt, and then we planted into both sites. And we also did a follow-up planting a year later, again with some seedlings planted into the burnt areas and some into the unburnt area. So this was part of the full translocation of this species. And the results from this experiment were really quite clear-cut. Survival was significantly greater in the burnt area. Even the second year after planting, second planting a year after the fire, this showed a similar pattern, although the survival effect was halved. So in addition to survival, growth was also greater for the seedlings planted into the burnt areas compared to those planted in the unburnt area. So you can see the small seedling down the bottom there that was planted into the unburnt area compared to the really tall seedling that was planted into the burnt area. So given these really promising results, this is an area of research we'd like to investigate for other species that recruit post-fire, but it certainly means that for this species, we would be looking at just implementing this on a wider scale for translocations and not worrying about planting into unburnt areas. So the experiment sort of was really helpful in guiding us in how we do our translocation. So moving on to the planting aspect now, I'd sort of like to highlight how useful the checklist for planting day is that's provided in the guidelines. They're really useful in making sure you have everything you need for successful planting, a successful planting day or days. One of the things to consider when planting is how the plants are going to be laid out at the site. So there are many factors that can influence this, such as the preferred microsites for the species, such as whether they're under vegetation or out in the open, and also whether you have an experimental design that you need to consider. But another factor to keep in mind is how easy it's going to be to find your plants again several years or decades after planting. In the case of this Cynathia down here on bottom left, we could find the plants in the open area easy enough, the ones in the foreground, But those in the bush at the back of the photo were very difficult to find as the vegetation grew up, and we frequently could not find the plants to monitor as time went by, making for a really patchy monitoring data set. So it's just another thing to take into consideration when you're thinking about where you're going to put your plants at the translocation site. Other factors to consider is adequate communication with the staff and volunteers on planting day. It's important to make sure that things are done the way that you need them done. So for example, in that top photo there, we had a miscommunication with staff about laying out weed matting at one of our sites. That meant that instead of using the weed mat pins to hold down the matting, someone instead chose to use an easier, or what they thought was an easier option, putting clods of dirt onto the matting, resulting in hundreds of dollars worth of matting becoming infested with weeds and completely wasting all our matting. So the communication on the day, or indeed just beforehand when you're trying to get the site laid out, is incredibly important to make sure that things are done the way they need to be done. Hygiene is going to be important to keep on top of during planting to ensure that weeds and pests are not introduced during planting, and it can be difficult when you've got lots of people on site. So you might have to introduce things like scrubbing down of footwear and equipment. So the example is in the middle photo here, where everyone entering site had to walk over mats and have their feet scrubbed down with a solution of bleach to make sure that we didn't introduce Phytophthora into our site. And the other thing is reiterating again about the importance of tagging or marking your plants in some way so you can keep track of individual plants and the information where they came from. And this may become far more important than you realise later down the track if things like taxonomic changes occur. But it's also important in just keeping track of survival and other aspects of plant health and growth over time. You'll need to think about the maintenance of the plants and the site after you've done the planting. You can do this as an experiment, so you can work out what is important and what is not needed. But you could also just do it on what you feel is the best option to do at the time. So I've provided a few examples there of setting up watering systems, having fencing installed, the mulching that we employed at some of the sites. But there's a whole range of other things you can do in the site, that's very specific to your plant or to the type of site that you've got. And you'll need to think about that at the time, about what you think might be important to your plants. And after planting, you'll need to monitor. What you're going to monitor will depend on what you'd like to know. So you primarily would like to know whether your goals for the translocation have been met. So for example, have you created a self-sustaining population? And there will be some steps along the way, or in other words, your objectives that you would like to see met in order to understand whether you've achieved this goal. For example, maybe you'd like to see 90% survival of the plants 1 year after planting. So these are the types of things you would think about monitoring. It's all determined by what you want to know. And the types of things that are monitored are many and varied, but there's lots of things you can consider monitoring in order to evaluate the success of your translocation. So frequently they include things like survival and establishment. Sometimes they might include growth, reproduction, such as flowering, fruiting, production of viable seeds. It might be the time to first reproduction, or recruitment of a second generation, so whether they're seedlings are produced or whether there's any asexual reproduction occurring. And then also things like survival and growth of second generation plants, and when they get to maturity as well. There are other aspects you can include, including monitoring for genetic diversity or whether there's effective pollination, but these are all going to be determined by what you want to know about your translocation. And it's really important to think about monitoring these over different time periods. So you're having short, medium and long-term goals, so you can assess different aspects of success over different timeframes to really get a good understanding of how successful your translocation is. So the monitoring should be species specific because there may be conditions that are important to the species that you need to understand. So for example, the Wollemi pine, which I know Heidi's going to talk about in far more detail later on today, but they wanted to understand about the influence of the different light levels on plant growth and survival. So they measured light growth along with things like plant survival and growth, because that was something they needed to understand. The other example here is of Cyanophia quartzitica, which primarily reproduces through underground roots, underground root suckering, so not seedlings. So in this case, we were looking for that clonal reproduction rather than necessarily seedling production as part of our monitoring program. The timing and duration of the monitoring depends again on what you're monitoring and what you would like to know. So for this occasion, we're looking for flowering and viable seed production, which meant twice yearly visits. So firstly at flowering in May and then seed release in November. We were looking for seedlings for the grevillea in the top photo. So we were looking several times in the winter and spring following a wildfire to understand whether this had occurred. And along the way, you'll need to evaluate whether you've met your objectives. You can incorporate the evaluation along the way into ongoing work. So thinking about what your monitoring is telling you along the way and whether there's any new questions you need to ask along the way. Be consistent in how you're monitoring to make sure that you don't sort of end up with gaps in your data set as best as you can. And think about how your monitoring results can be incorporated in ongoing work, or in other words, do they trigger some sort of management response, such as changing the water regime or now starting to control the weeds? This is the sorts of things we consider as adaptive management. And can the results be used to help in planning new translocation work for that species or for any other species as well? So moving on, I'd like to give some examples from our work here in WA about some of the different types of monitoring of translocations. So I'm going to talk first about how we use the monitoring of many translocations to look at how effective our watering and herbivore exclusion treatments are. So for a number of our translocations, we've watered the plants for the first summer or two following planting, and we've also fenced the plants or entire translocation sites to protect them from herbivores. We've now used the monitoring data from 53 species from 60 sites planted across the southwest of Western Australia and monitored these between 1998 and 2017 to assess how important these techniques are for improving survival, growth and reproduction of translocated plants. So we found fencing had a consistently positive impact on survival across all 53 species. I've just highlighted, you can see the effect size there with the red arrow. I haven't shown the results for growth or reproduction here because it would just make the slide a little bit too complicated. But curiously, the fencing didn't have any impact on reproduction or growth and had a slightly negative impact on crown growth as well. But for the survival results alone, it makes it a very worthwhile investment. Also, large fences have the added advantage of protecting any seedlings that recruit naturally too as well. Watering over the first 2 summers following planting had a positive effect on survival for some of the 53 species, but not for others, but overall the effect was negligible. However, again, I haven't shown the results here for survival and, for growth and reproduction, but it did have a consistently positive impact on survival and, growth and reproduction for all of the species. So pretty much the reverse of what we found for fencing. So despite the negligible survival response across all species, we had such a consistently positive growth and probability of reproduction following watering, and we'd probably still view watering as a useful bet hedging strategy given the rarity of these species. Sorry, I didn't show these. I mentioned another aspect that you can monitor is genetic diversity and pollination, and we've done this here for a species called Lambertia orbifolia. There's 2 subspecies, one called Lambertia orbifolia subspecies orbifolia. It's known near Albany, is listed as critically endangered, and it's got 2 translocated populations, and the other subspecies, Lambertia orbifolia Scott River Plains, is known near Augusta and is endangered. It has 1 translocated population. We found that genetic Lambertia orbifolia subspecies Scott River Plain, which was established using a single source population, had similar levels of genetic diversity to wild populations, and we also found that Lambertia orbifolia, which was established using several source populations, had genetic diversity that's higher than wild populations. We also found that the inbreeding coefficient is similar between translocated and wild populations of Lambertia orbifolia Scott River Plains, but this measure was elevated in both Lambertia orbifolia translocations compared to wild populations, and we suspect either the Weiland effect or the ex situ propagation of seedlings may have played a role, but this effect should hopefully dissipate over subsequent generations. We also used the genetic data to do a structure analysis. The analysis supported here the taxonomy of the 2 subspecies, but within each subspecies there were 2 different patterns. Lambertia orbifolia Scott River Plains showed little differentiation between wild populations, with the exception of 1 small population, the one shown in green, whereas within Lambertia orbifolia there was a lot of genetic structure, with each wild population genetically differentiated from each other. So understanding the genetic structuring within a species can help with understanding translocation, planning. In the case of Lambertia orbifolia Scott River Plains, the translocation on the end was established using a single source population, but has some evidence of gene flow, either historic or current, between the two population cluster. This lack of structure between populations suggests using a single large healthy population was sufficient to capture the genetic diversity of the subspecies. In contrast, for Lambertia orbifolia, two different source populations were mixed together to establish each translocation site. Because there is structure between the populations, this strategy of using a mix of source populations was advantageous, as it increased the genetic diversity at each translocation site relative to the wild populations. When we investigated the mating systems of these two subspecies, we found the patterns of pollination were similar between translocated and wild populations of Lambertia orbifolia Scott River Plains. However, although some mating systems parameters were similar between translocated and wild populations of Lambertia orbifolia, the levels of inbreeding were significantly higher, highlighted in the bright red circle, suggesting that pollinator activity is not optimal, and the increased level of genetic diversity that we captured during translocation establishment may not be maintained into subsequent generations. So therefore, we will need to monitor the mating system values in subsequent generations, and if we still find suboptimal pollination, we will need to consider what we can do to improve the situation. So these studies have given us a fair bit of information about how well we did in the translocations and what we need to do in future translocations. A different aspect to monitor is how resilient these new translocations are to disturbances. This resilience is important if these new translocations are to persist. So we have monitored the response to one of the major disturbances, which is fire. In some cases where the fire is planned, as in the photo on the right, but more often when the wildfire occurs, as in the example on the left. The Grevillea maxwellia is an example where we monitored after a wildfire. Our plants were only 2.5 to 3.5 years old before the wildfire occurred, and only 2/3 of these had flowered. When we heard there'd been a very hot wildfire burn through the site, we expected the plants would be killed because the species was a cedar, and that due to the young age of the plants, there may be only very limited seedling recruitment. But in contradiction to our expectations, 78% of the plants re-sprouted following the fire, and 98% of these had flowered. Have now flowered. There's also a small number of seedlings recruited. And in the photo at the bottom right is the plants earlier last year, all looking really healthy. So the response of the plants to the wildfire was a positive sign that this translocated population is resilient to disturbance and one indication of a successful translocation. In the case of Acacia cochlecarpa, subspecies cochlecarpa, previous research showed the species primarily recruited after fire. So instead of waiting for a wildfire, we applied a prescribed fire to one of our translocated populations to see whether it had the ability to respond to each disturbance. If it did, it would then meet one of the criteria for success we had set for this translocation. So following the fire, we found that 6.5% of adult plants escaped the fire, showing the fire was patchy. 2% of adult plants re-sprouted, something we had never seen before in this species, and a large number of seedlings recruited. We continued monitoring the response of this population and found 77% of these seedlings survived the 2 years post-fire, and that a quarter of these had become reproductive within those 2 years. So the population was able to successfully respond to fire and recruit a second generation, which is now reproductive and meets our success criteria. So I shall finish here with a quick thanks to all the people who've worked on plant translocations that I've talked about, to Peter Vesk and Jian Yen for their work on the analysis of the plant translocation data, and to Heidi Zimmer for some of the slides on the monitoring and evaluation. Thank you.
Thanks, Leonie. I was just busily typing you a question. It's really nice to have an overview of all the potential moving parts of a translocation. I know some of those tactics seem obvious, but also I think they're the kind of things you only learn once you've made the mistake. All right, now next up we have Dr. Linda Parker from the University of Melbourne. Linda, you can get on screen. Now, Linda's going to be giving us a case study of Astelia australiana translocation in the Otway region and some of its unexpected findings. We have a little video that Linda would like to play first, so that's going to start now.
Can you see me?
Yep.
A lot of people don't know that tall Astelia exists, in part because it's a rare species. It's a very cryptic species as well, in the sense it's hard to find. It's only known from very few locations around the state of Victoria, most of those in the Central Highlands. And in the Otways, this site right here is the only place that does exist.
So before I began the project, I didn't know much about the species. I'd seen it once and just thought, you know, it's a nice understory herb, but it doesn't do much. We had one site burn in 2009 in the Black Saturday fires. There was 4,000 plants estimated to be there before the fire, and after the fire there was only about 1,000 remaining. But one thing we did notice, what was neat in this site that burnt, was where the canopy had been removed, so the fire had gone straight over the top and not really burnt the understory. We had mass flowering of the Astelia because they've suddenly got a whole lot of light.
At all the other sites where there was no wildfire, we had hardly any flowering. And where we did see flowering, it always happened to be in really light areas. And that's where we came up with the hypothesis that, hey, maybe there's something with light. Maybe light is a really important mechanism in this species' reproductive ecology. And so we set out testing that. And what we found, we don't have to do much. Remove one tree, maybe remove a tree fern. That's all we need to do. You know, it's a very limited amount, going from 1% light to 4% light availability. That seems to be the magic number with this species, is what our analysis suggests, 4% light.
Previously, we thought the Astelia was just like a large lily. It rarely flowered or reproduced, and we just didn't know much about its ecology. But now we know that it does reproduce. It can reproduce every 4 years, and we know that the edible berries are taken by a whole suite of birds and mammals like eastern spinebills and pygmy possums, olive whistlers and lyrebirds and echidnas. The echidnas love it, the ants, the swamp wallabies love the nectar and stuff. So it is part of the community. It provides habitat, it provides food. It just needs a little bit of help to get some reproduction occurring, and then it gives back in so many ways. I wanted to thank Fleur and Chantelle and the ANPC for inviting me to present today. Following Leonie, I feel like I really don't have a lot of translocation experience, so maybe I'm a bit out of my depth. But I wanted to share with you a little bit of research I've done originally as part of my PhD into the tall Astelia, Astelia australiana. This species occurs in Victoria, in the Central Highlands and the Otway Ranges. It grows in cool temperate rainforest and riparian vegetation, and I think it's a pretty neat herb. And as you saw from the earlier video, it was just something fun the ABC picked up on. We've done a little bit of research into its ecology to try and understand why it's threatened, and I'll start with that. A bit of a background, and then I'll go into the translocation work that we did. So this is a slide showing the current distribution. Like I said, it's in the Central Highlands, and there's about 14 sites in the Central Highlands. I've marked them with a black dot, and then I've shown translocation sites in yellow. And then the Otways Ranges is just one known site. That's down in the bottom corner, and then we had a translocation site there in yellow as well. So just as a background, we know that the species is trioecious. It means it has female hermaphrodite and male flowers. We can also have the hermaphrodites can be have male and hermaphrodite flowers on them, or female and hermaphrodite flowers on them as well. When it reproduces, it forms 3 clones. So that's vegetative reproduction, and at the same time it will produce a flower stem out the centre, and that has the inflorescence on it, and then the females and hermaphrodites will produce seed. And you can have a variable range of seeds. We found between like 1 and I think we had up to 15 in some fruits. And following dispersal, which happens through a range of species, I've got some photos in the next slide, you'll have germination, and then it takes approximately 4 years to form like 3 to 9 leaves. It's very slow growing in the understory. The 3 ramets that are formed from the clonal reproduction, they can go on to reproduce after 4 years their own and start that cycle again. The species, as we said in the video, it was thought to flower very irregularly, and there was low reports of flowering, so we thought most of the reproduction was clonal, but now we know that they both happen at the same time, but I guess successful reproduction is mostly from the clonal growth, because the seed germination success appears to be really low. And these are some of the pollinators and dispersers. We've got pygmy possums, lewins, honeyeater, swamp wallabies with the nectar, so potential pollinators. The silver eyes love the species. Yeah, and some of the threats. So there is a national recovery plan for the species. It was previously, when I did my PhD, it was listed as vulnerable. That's been uplisted now to critically endangered under the FFG Act. We estimate there's 10,000 individuals, and in 1993 a monitoring program began for the species. That was by DELP. Vivian Turner was the main person involved with that. And as part of my PhD, 20 years on, in 2013, I monitored all those populations that they'd initially put in the monitoring program, which was all unknown populations, and found 57% decline of individuals in those populations. A further 5 years on, after my PhD in 2018, we went back out and surveyed again and found that the species had declined by 68%. And at the end of this year, in summer, we survey in summer because that's when they're flowering, and so you can see the sex of the species and the flowering rates. We'll have the 30-year monitoring. So some of the implications was that it required an uplisting for the conservation status, which it now has. The current seed storage techniques appear not suitable. So we've done some trials with Andre Messina at the Royal Botanic Gardens, and they were previously storing them at -20 degrees, and we found that the seed's somewhat recalcitrant and isn't viable with those storage conditions. We've also tried just storing it at 4 degrees, which is not a really good long-term option for any seeds. I don't think it's suitable. And 2 years past storage, it's very low germination. None at -20, and -80 is showing a little bit of promise, but it's really low viability of seeds in long-term storage. So we decided we could do a trial translocation, which I'll talk about shortly. And then following the success of that, we've done another translocation. Then we've done some genetic analysis and some population viability analysis to inform future translocations. This is a picture of a sandbar, because herbivory by deer is impacting a couple of the sites. And there's also a picture of some burnt trees. We had one really large site that I mentioned in the video, had 4,000 individuals, and then after Black Saturday fires, it was down to 1,000. So we lost a lot in that site, but it did encourage mass reproduction because of the extra light. So from that hypothesis that, you know, they need the canopy decline because reproduction takes a lot of effort, takes extra light that they're not getting under the rainforest canopy. We designed a manipulation experiment where we created canopy gaps and had some full canopy controls just next to it within 4 sites. And we had flowering in 8 of the 12 canopy gaps after 2 years. So as part of the PhD, we did do a trial translocation. There's a couple of photos of my supervisor, Craig, and 2 colleagues assisting with the translocation, Tim and Ben. So we moved 54 individuals into 3 site types. So that's 18 into each. The control was we dug them up and we literally put them 50 meters away, just to see if it was like the shock of being moved would be detrimental to the individuals. Then we had a locally absent site, which is where they're walking to. It was just down the road, and then we had an absent site. So that was about 5 kilometres away from this source site, and that actually had the most survival. This was survival was measured 2 years post translocation, and we had 89% survival, which we thought was good. In terms of success rating, we weren't looking for reproduction yet. We did do some micrositing where we made sure that we planted them with sufficient light in areas that didn't appear to have predators such as deer or disease. We made sure we used, it was very simple to get the plants out. They could almost be pulled out. It's very muddy site, but we did use a shovel to avoid damaging the rhizomes. They have very few roots and can grow as epiphytes. But yeah, we moved them out with the shovels. We sterilised the roots with phytoclean diluted to make sure we weren't moving any disease, but we were very confident that where they were currently was disease-free, because if it wasn't, we know that Phytophthora is an issue, and so is Pythium, and results in the very quick death of individuals. So we're very confident that what we were taking from was disease-free. And that work, the trial translocation, then led to a larger translocation post-PhD. That's published, the ANPC journal, and also in a IUCN as a case study in their global conservation translocation perspectives. If you want to read further, we basically moved 200 individuals and had 2 different site types. So one was a reintroduction into a site near Gembrock, where we've fenced because they basically only had 2 individuals remaining, and that site had been hit very hard by deer. Also had some disease. So we fenced the area around those 2 individuals with the help of DELP and Melbourne Water, and then we planted into those ones as sort of a reintroduction, I guess. There was still 2 left. And then the rest of the sites were to create new sites just further out. I'll show a map shortly that shows where they were. It's basically, they're very closely connected within the Central Highlands, the sites, and we want to minimise the risk of one particular wildfire, I guess, taking out a whole lot of sites like it did take out at the one large site. I also did a seed and seedling translocation trial. It wasn't very successful, so I won't talk too much about it, but basically it follows on to what Leonie just said. It's really hard to find these things. There's so much leaf litter in the understory that the seedlings get lost very quickly, and they're so slowly growing. The seeds grow very quickly in the nursery and readily germinate when they're fresh. So that was a good thing, but in terms of planting them out, we had 0 survival after 2 years of seeds and seedlings in the sites. Following the translocation, we have done some genetic analysis, which ideally, yes, as was said last week in the symposium, it would be good to do at the start. Basically, we wanted to know, is there gene flow occurring between the sites, where we should put the populations to encourage gene flow for the new ones? And are there any sites that need a bit of assistance in terms of receiving individuals to increase the diversity? And where should we take the plants from to get the best genetic diversity within the new sites? And so the genetic analysis was done after the second translocation. So it's now going to be used to inform any future translocation. This is just the summary of what we found. But basically, if we look at all the sites, including the Central Highlands and the one in the Otways, there's 5 groups identified based on the differences between the genes. And if we just look at the Central Highland sites alone, there's 9 groups. So it's a bit of a summary. Here's the Central Highland sites on the map. it shows the 5 groups, but it's a little bit hard to see, I guess. Basically, what we found was, the 9 groups, that no one site needed rescuing from the genetic data. They all seem to be quite stable like that. The current translocation sites that we've got marked in yellow may be a bit too far away to expect natural gene flow when they do reproduce. And that's all I sort of focus on here and go back to the translocation, except to say that the Otway site, it had been previously reported in the literature that it was expected to be just a sterile site. There's only 150 plants remaining there. It's actually less than that. And from the genetic analysis, we found that it's more diverse than all the Central Highland sites, which is a really interesting result and will help us to decide what to do in terms of increasing the genetic diversity of the species. And that just shows you the little map there shows that there's only one current known site. We've translocated it to a site in the south, and the other sites were, there was historic sites further north that were hit by fire in the 1920s, wildfires, and they no longer exist there. And so we identified from some species distribution models potential new sites where we could translocate into. And then obviously we have to make sure they're disease-free and herbivore-free before we do that. So the implications of the genetic work on translocation were that sites that we form from now on need to be within 2 kilometres to expect gene flow between them. The Otway site is important genetically, and it should be used as a source population to increase the genetics. We haven't quite worked out what that means for the species in terms of where we'll put them and what the best mix is, but yeah, we will use those results and our species distribution models to guide that in the future. I wanted to take a minute to say that I've only done 2 years of monitoring post the large translocation, and we had 22% of the plants die after 2 years, which was 56 plants. And I think we've only measured success based on survival, but into the future we would like to, monitor this further. It's ongoing, but we need to, I guess, arrange funding for it is part of it, but also we'd like reproduction to occur in those individuals, and so that's why we've looked at the light dynamics when we're placing them in there, and we expect that it will happen, but it can take a few years for that to happen. So survival is our only success measurement at the moment. Growth was similar between all sites, and we did look at that. I think that's about all I wanted to say, except that it's monitoring and evaluation is ongoing, and that was it. But I have judged the trial as a success. 22% decline in 2 years is pretty high, but I feel like that was probably initial shock, and then those ones that have persisted will continue, because that's what the species does. It just is long-lived, more than 30 years in individuals, and I think it's just quite happy to sit in the rainforest and do nothing, not necessarily reproduce, just wait until a canopy gap forms, and then it does its business. But the species is important for other individuals in the community. It provides food, it provides shelter, so it's a good habitat. And I think these areas that we're reintroducing it to possibly had it in the past. We just didn't know, because it's been declined for whatever reason, or else they were just further beyond its range, as in more than a few kilometres, and it hasn't had the chance to disperse to those areas. But they look like ideal habitat for the species, and that's about all I have to say.
Thanks, Linda. That was really, really excellent. I'm pretty thrilled to see that this project has continued and expanded over the decades. I know a lot of it's done pro bono, but you've done an awesome job with it. So thank you for giving us guidelines to move forward. Now, our next speaker, unfortunately, is not able to join us last minute, but Shannon Dwyer from Gippsland Water is going to be jumping into that slot, and then you get to learn about my work, which I didn't expect to talk about, but there we are. Now, last week you saw the orchid translocation video that Shannon featured in, but actually Gippsland Water is involved in quite a few different conservation translocations, and Shannon's going to be explaining what they do apart from managing water resources and the role that companies can play in conserving species. So Shannon, if you're there, you can jump on and get going.
Yeah, thanks everyone for having me. Just wanted to share a bit of a story on translocating the Wellington mint bush. And yeah, as Chantelle said, we're a water authority, so it's not you know, our core business, but it's something that we take pride in, and yeah, just part of what we do. So yeah, Gippsland Water's based in central Gippsland in Gunai and Bunurong country. We supply around 66,000 customers, both potable water and manage the sewage from those customers. Manage 14 wastewater treatment plants, 15 water treatment plants, and almost 4,000 kilometres of water and wastewater mains. But my job, some of which my job, you know, includes managing some of the risks from those and managing the environment, but also conservation is a big part of what we do in the environment team. So we've got around 6,000 hectares of native veg across our region that's both freehold and crown land vested in us. Some of those are formal conservation reserves for biodiversity offsets. We also have carbon offsets, but quite a lot of it, the vast majority is native veg with no specific protections apart from just being natural vegetation. So yeah, we undertake weed control, pest animal control, those sorts of things, but also where required, we do specific management of threatened species and on occasion translocation projects. So over the past 5 years, we've done 2 translocation projects for threatened species. As Chantelle mentioned in the video last week, we did the Thalamitra epipactoides, which occurred nearby our property, but there were no records on our property down at Dutson Downs, but we set up a translocation site there and did that a few years back. And we also have a small population of Prostrantha galbraithii, Wellington mint bush on our property. And so that's the focus of this presentation. So Wellington mint bush, it's a small shrub, or can be sort of medium to tall shrub occasionally, around 2 meters, maybe 3 meters when you get a big bugger. It occurs only on the Gippsland Plain and only within a very small area of the Gippsland Plain, on old sand dunes through the sort of eastern part of central Gippsland, I guess. Around the lower area of the Gippsland Lakes and inland a little bit. Vulnerable at Commonwealth and endangered at Victorian level. It has a lifespan of around 20 years, give or take a few. Some of the stranger things, not stranger, but difficult things, I suppose, for the conservation of this species in particular, is that the germination of the soil sorts seed is only really happening after fire. I'm yet to find any recruitment in areas where older plants are without any fire going through. The seeds are quite small. They're produced basically every year. There's only sort of four seeds within the calyx that are produced, and they drop out every year. And they're probably only moved around by ants. They're so small, they're not really a food source for anything larger, and it'd be difficult to find amongst the litter. And the other difficulty is that the seed dormancy of this species is particularly difficult, given that it's triggered to germinate by fire. It's one of those things that, you know, it's not quite known whether it was smoke that's germinating it, or it's at the heat, or some other element of the fire. So it's another difficulty with propagating this one and moving it around. So why this plant? Well, as I alluded to before, it's very limited in its distribution. This is the entire distribution of the plant within the world, which is just Holy Plains State Park and Dutton Downs over here to the right. Because of the distance of these populations, so they're around 40 k's apart, there's very, very unlikely to be any gene flow between the two. They're just bee pollinated as far as we know. So there's really no movement of genes there over the last, any sort of recent history, perhaps the last 100 years. Quite a bit of land clearing in between the two populations, a lot of plantation, that sort of thing. So there's not really any connectivity or other known plants in between to create that gene flow. And so within Dutton Downs, we've got the smaller of the two populations. One of the within our population, there's two smaller ones, and one only has 15 plants in it, and the other's about 150 plants. So given the small number at Dutton, it's a very real risk of local extinction there without any active management. So because of all those reasons and the risk of local extinction, we decided to trial a propagation and translocation of the species, and collected seed in December 2017 from the Dutton populations, both of them. And we germinated the seed using a local nursery and relied on a method that was developed by Arthur Ryler Institute a few years back. So they did a bunch of trials using smoke, heat treatment, and gibberellic acid treatment. And turns out, as seems to be the case in a lot of things, gibberellic acid did the trick best. So we decided to rely on that rather than fiddling around with smoke treatment and that sort of thing. So we ended up getting about 250 plants using that method, and we're able to grow those on for translocation. So as part of this project, we applied to DELP and were successful in getting a little bit of funding to help with the propagation and the construction of some cages to protect the plants when they're in the field. As you can see from these photos, that they're not particularly elaborate in their design or construction, but very effective. So one of the risks for translocation or even just plants in the wild is browsing, and not just from deer or any sort of pests that get in, but wallabies, kangaroos, native animals do browse on these things for some reason. They're difficult to find in the understory, but amongst the heathy woodland that this species lives in, there's not a lot that's very palatable. So this is probably one of the few species that they stumble across and get a munch on. So keeping out the native fauna and the pest fauna was critical to making sure this was going to be a success. And so the design of these cages, there was a bit of thought put in, although they're pretty basic in their construction. The idea was to make these cages big enough to be able to fit a few plants in, so that we can create some sort of small populations within these things and hopefully get a bit of cross-pollination between plants, but not so big that anything that could jump the fence, because they're not particularly high, only about waist high. Anything that could jump the fence wouldn't want to, because it might get stuck in there, and it's proven pretty successful. We haven't had any wallabies, deer, or even wombats getting into these with the skirt put around the bottom. That's stopped any diggers getting underneath. So for the project, we established two translocation sites. Now, as I mentioned earlier, one of the key reasons for this was the risk of extinction of this population from low numbers, but also for the population being concentrated in this small area of our property. So at Dutton Downs, we manage about 8,500 hectares, which about half of which is native veg, and the other half is wastewater lagoons, some organics processing, and farmland that we reuse wastewater on. So there's quite a diverse mix of land uses down here. But I suppose the main issue with this species was it's contained to this one area, yet there is suitable habitat right across the property. So in terms of reducing risk of extinction, we decided we'd put in a couple of populations, attempt to establish a couple of populations in different areas of the property, disconnected from the current populations, and in the hope that we could establish some new plants, create some seed store within the soils, and if we do have a wildfire through at some stage, that the risk is minimised, that we'll lose all plants in one hit. So this project became more important in late 2019 when there was a large wildfire that ripped through Holly Plains State Park, so the other larger population of this species. And throughout that fire, almost all of the smaller populations of the species within the park were raised by the fire. Now that's not a major issue, but we didn't know how they were going to respond. It was a fire in the middle of summer, quite hot, so could have consumed the seed in the soil. So for a while, the Dutson population was thought to be the only population of the species left. So now we're talking about less than 200 plants potentially left in the wild. So the success of this translocation project became a bit more important to make sure we could make it work. So after a little while, though, it was good to see that not all of the plants in Holly Plains were consumed. There was, I think, at least one population that remained unburnt. And by October, late October, there was reproduction seen in the soil. So we had plenty of seed popping up. So good news that the Holly Plains population is doing well. As far as I know now, there's many, many more plants than what there were before. So that's great. So in June 2019, we undertook our planting. So that was 2 years after seed was collected. It was propagated early in 2018 and grown on throughout 2018 for planting in June 2019. And as I said earlier, we did that across 2 sites on the property. Collected some basic information when we were planting, so tagged all the plants individually, numbered them, and collected heights to work out, maybe determine whether bigger plants were more successful or smaller plants were more successful, so we could learn a bit from this translocation to take into the next one potentially, or any other work with this species. As part of the translocation, we haven't done any really significant management interventions. So no watering, no fertilizing, nothing else you can think of to sort of nurture the plants along. It was very much, I guess my motto on some of these things is if they're not going to survive without active intervention and spending money and resources going out planting these things, then we need to think about a better way of doing it, and particularly being a water corporation, it's not our core business to be out there watering plants. So we want to make sure they're successful and set them up for success, but don't want to be locked into any ongoing watering programs throughout summer and that sort of thing. So really the only intervention that we've had in these is hand weeding a little bit of bracken now and again. It tends to get pretty thick in there without animals passing through. So whenever we're out there doing the monitoring, we'll pull out a bit of bracken and open it up for the plants, but otherwise pretty much maintaining the fences and that's about it. And each year we go out and do that monitoring, which we're collecting basic information, not a great deal, but heights of plants and whether they're flowering or not. And then later in the season, we haven't done any specific checks on the seed and logged that against each plant, but we are seeing seed formed on these plants. So the results from the translocation are kind of mixed. You can see here we've lost a significant amount of plants. So yeah, as at last year's monitoring, there was just 108 plants left out of the 222. So, you know, by anyone's measure, that's not hugely successful. But the plants that are there are doing really well. The vast majority of them have flowered, 92 out of the 108. So 85% of plants after the 2 years have flowered. And like I said before, we're commonly seeing seed set in these plants as well. So some of the perhaps obvious, but I think important results to take out of this one were that plants that were larger when they were planted flowered earlier and have a higher survival rate. That might be because this is just a theory at this stage, but because the population's so small, we had quite a few runts, I'd call them, in the group of plants that we planted. So maybe a little bit of inbreeding depression is leading to less fit plants. So some of those runts that were smaller probably haven't survived through this first couple of years, but the larger ones that were planted have done better, so they're probably fitter and more vigorous plants. And there was no significant difference in survival or vigor of plants between the two sites. So both sites were found to be suitable, obviously, for the species to be re-established there. Now, as was mentioned earlier, some experimentation was done with this translocation as well. So not just these two sites, but we actually kept a few plants from the original 250 that were propagated and potted them up and grew them on a bit bigger. And the following year planted them in some atypical habitat. So what you can see here is 4 plants that were planted into a wildlife corridor that we've got for another species that we manage, which is the New Holland mouse down at Dutson. We've got a wildlife corridor that we've been doing some revegetation on for quite a while. And so it's a reasonably secure site, but it's dominated by pasture grass and is periodically sort of being revegetated. So we established a few plants in here just to see how they'd go with being in an open, windswept pasture, and they're doing really well. A year on from planting, they're flowering, they're quite vigorously growing, even though they're competing with the grass in here. There's recently been a bit of hand weeding done in here, but other than that, there's no real management of these. And the other experiment, and I'm first to admit these weren't particularly scientific experiments. They haven't been, you know, overly designed or anything. This was just a bit of we'll see what happens. The second area was in typical habitat, but with no cages, so separate from the other two sites. Another location, very similar habitat to where the species, the parent population is, but we put no cages in. And very, very quickly, the local fauna had found these ones and decimated them. And there's just a few left now that are sort of struggling, as you can see there. So, yeah, perhaps that result was pretty obvious. We've seen them be, even the taller plants get munched, but you know, it was worth a try just to see whether we could get some establishment in an area where animals might not have known that the plant would occur, but they found it anyway. And so the other site in the pasture gives us a bit of promise that we might be able to include this plant a bit more broadly in revegetation efforts around the property, and maybe we don't need to be so careful in our site selection and matching the habitat, you know, exactly to where the parent population is. We can probably plant these things a bit more broadly around the property and hopefully get a bit more success. Thank you, everyone. That's it. I'll hand back to you, Chantelle.
Thanks, Shannon. I really liked your comment then about runts. Coincidentally, your next speaker is going to be talking about runts, and that next speaker is me. Now, I thought I had got away with not giving a talk this time, but unfortunately, because Kate wasn't able to attend, I'm going to be giving you a very quick rundown of the work that I do, and I'm going to rely on somebody to tell me if you can't see my screen. I'm assuming that you can. All right, I'm putting on a stopwatch. So the species that I'm going to talk about is Hibbertia spinantha. I'm not going to be talking about translocation, but I'm going to be talking about some of the ecological considerations prior to translocation. but we needed to look at to hopefully make sure the conservation planning of this species was effective. Now, this species was discovered in 2007, but it wasn't named until 2015. It's known from 4 populations, 3 right in the Sydney Basin and then 1 kind of adjunct population up near Maruta. 2 more populations were found last year, which was exciting, but they're not included in this talk. Now, I started working with this species in 2015 before it was even described as part of a failed mitigation translocation where we had to be augmenting an existing population as insurance for a big road development. And that translocation failed completely. So I decided I wanted to do a PhD to find out some of the ecological factors of the species that would help increase the chances of it surviving and having a successful conservation in the future. If you want to read about my horrible failure, it's in APC. That's always good to share failures, right? So the things that we didn't know about this species were pretty much everything. Breeding system, we didn't know if it could self-pollinate or if it required pollen from unrelated individuals. We didn't know if pollinators were present. We didn't know the genetic health of the populations. We didn't know how to propagate it and to make sure it survived. I've done that work, but I'm not going to talk about it in this presentation, but please get in touch with me if you want to have a chat. And then basically, I wanted to use that information to find out what conservation actions we should be doing. Should we be augmenting populations? Should we create new ones? Should we have an ex situ collection? So the first thing to look at is the breeding system. I hope you like these little GIFs. We looked at 4 different methods. So 2 different breeding systems are orthogamy and gytonogamy, and they're both selfing. So on the left, you can see orthogamy, where you have pollen from the same flower. Gytonogamy is pollen from different flowers on the same plant. And then outcrossing is when you have pollen transferred between other plants in the population. I did 2 different outcrossing treatments. One was open pollination, where basically I let the pollinators, if they were about, do their job. And then the other one was I transferred pollen between flowers in those 3 close populations. We couldn't do the 4th one because that's on private land. And I used an electric toothbrush. You can see a video there. I used an electric toothbrush to mimic a buzz pollination. I won't get into the semantics of anther structure. Suffice to say that Hibbertia in theory buzz pollinated, and part of the reason is that for that porocidal anther, the holes in the top of the anther require vibration. Yes, so as I said, we transferred pollen from the ride population to the 2 other populations and then between them as well. And basically we found out that this species is an obligate outcrosser. So that means that we effectively got no seed from any of the selfing treatments. So it can't pollinate itself. We got significantly more seed from open and outcrossing treatments. Okay, so that's answer 1. It can't pollinate itself. Then we needed to find out what pollinates it. I wanted to show you this little setup of mine. It's very simple and maybe you can use for your work. Just a phone app, Salient Eye. It's a home safety app with a little mannequin holding it to get recordings of pollinator visitation. And basically we found out that the primary pollinator is Lasioglossum or sweat bees, which incidentally do buzz pollinate. You can see one doing a lovely little job here. And actually it's doing some gytonogamy there, which is now we know not very effective for the plant, but very effective for the bee. Also syrphid flies and bombolid flies also visited, but irregularly, and they just collect individual pollen grains. I wanted to show you this. These are some shots from the Salient Eye app. It really worked better than I expected. If you have a look, maybe you can find what triggered it. And there you go in the red. I'm flashing them at you now. They're what triggered the app. Waving grass would also trigger it, which is very annoying, but luckily it was low enough that that didn't happen. Okay, so we know about its breeding system. We know that the pollinators are present. The final thing I needed to look at is genetic health impacting pollination success. So we did a pairwise kinship analysis, which Maurizio presented last week. What you want to take away from this is everything red is a clone. So basically we found out that all of the populations are highly clonal, not very useful for a plant that can't pollinate genetically identical individuals. So of the 71 stems, we found that 43 were genetically unique or genets. We redid the analysis, the pairwise kinship, removing all the clones, and we still found that most of the populations were highly related. So siblings or half siblings, which is denoted in the yellow and the orange. Again, not great for this species. But is it a problem? So I answered, is it a problem, in a couple of ways. We looked at seed fill. So seed fill is a proxy for viability. Unfilled seed is, you can see on the left there, and then this is a lovely example of filled seed. So filled seed is probably going to be a viable seed. Unfilled seed is not. And we found out for the Tarahumara population, which is the biggest, that outcrossing, so transferring pollen between unrelated individuals, produced significantly more filled seeds. Those aren't error bars, they're confidence intervals there, by the way. And the reason that we think for that is the Tarahumara population has a lot of big old clones. You can see them in that picture, and they are probably swamping the pollinator foraging in that they don't fly to unrelated plants, they just go diagonously between flowers. Another way you can look at inbreeding effects, and this is where I'm getting to you, Shannon, is germination. We tried to germinate over 200 seeds that I collected from those different treatments using smoke and a control. Found out that smoke significantly increases germination. Great. No impact on outcrossing or open pollination on germination rates. But when we look at the growth of the plants that germinated, of which we only got 41, not many out of the 200 at all, you can see that the mean size of outcrossed germinants is much, much bigger. So there's your runts, Shannon. Basically, it's a really good indication that inbreeding is affecting plants. So what we've planned, or what we've done already based on this information, is we've set up an ex situ clonal population, potted and in ground. And we have done an augmentation of the ride population, which had one single individual. So it was really eligible for an augmentation with the 41 seedlings that we germinated. This is me planting in optimum planting weather, very wet. If you look on my Twitter, you can see how wet I actually was. My kind of takeaway logistically was this took around 5 years to get this information. It involved a whole lot of collaboration, and conservatively, it cost over $200,000, $265,000, half of which at least was in kind. That doesn't include all of my PhD time either. Okay, so that's me. That's what I'm working on. Thank you so much for listening. I think we're almost on time. We're going to have a break now and come back at 2:05 when we have a Eureka Prize nominee coming to talk to us back at 2:05. Thanks, everyone. There's sort of this perception that scientists or ecologists know a lot about species. You know, we've done all the work, we've done all the research, we have all the answers, and we really don't. This is Hibbertia spinantha, or Julian's Hibbertia. And this is the plant that I have been researching. I haven't done 58 yet, have I? Yes, you heard right. Me, the narrator of this series. I am the voice of Plant Heroes. Except this is the voice of Plant Heroes. There are only 4 officially known populations of Hibbertia spinantha, and they all occur within the Sydney Basin.
In 2007, it was discovered in this particular place that we're at now. a council staff member came across a plant that he thought was something different. And through a process of scientific review, it was found that it was a new species. So it was listed as critically endangered in 2015.
I first came across this species when I was working as a consultant. As part of the conditions of approval for a big urban development, we were asked to augment an existing wild population with nursery-grown plants. But things didn't quite go to plan. We ended up with all of them dying. I was responsible for the life of this critically endangered plant. So I said, I know how I deal with this nervousness. I will do a PhD. The 4 populations are all in urbanised areas. One's on private land. One only has 1 plant in it. They're not guaranteed survival at all. That's not good at all. The single genetically unique individual in the population has died. They do that. They seem really healthy, and then all of a sudden they will just die. We can't be assured that the wild populations are secure. So what we decided to do was set up an ex situ collection. To do this, I needed a lot of help. So I started working with volunteers and staff from Hornsby Shire Council. We set up 2 ex situ, or away from the wild populations. This is a translocated population. So this population here is going to be irrigated for 1 more summer, and then it will be on its own. But these plants are far enough away from the wild populations that we can bring in all the genetic material, and we can have a little bit of a planted population and see how they go producing seed naturally before we start augmenting the wild populations. The second population was stored in the Hornsby Shire Council Community Nursery.
We're back at the community nursery now, and we're going to do all the preparation of the cuttings and setting of them in the striking mix with their plant hormone applied. Then they're going to go across to the glasshouse, where they'll stay on the heated benches and misting system until they've struck and made their own root system. So that's the entire Hibbertia spinantha population that's known.
The ex situ collection was an insurance population to give us time to plan for its conservation and answer all the things about its ecology that we didn't know. So first I had to figure out if they could be pollinated. So I sat around with my phone and made little camera traps with my phone with a home security app. I found that there were a few things visiting it, mostly a sweat bee called Lasioglossum. It's like, I think it's brushing the pollen to its back legs, and they are maybe not classified as buzz pollinators. So when I went, okay, they're probably buzz pollinated, to hand pollinate them, I said, how do I mimic a bee? So I got an electric toothbrush, and I just really tested, can I get pollen to be released from these anthers? And yeah, I did. The pollen just sort of exploded everywhere. And then I got to go and do that in the field. I was like, oh my God, I'm a bee. I needed to check if they could self-pollinate. And basically what I found, it's essentially self-incompatible. So it could not use pollen from the same flower to pollinate itself. It's my toothbrush. These populations are isolated now. There's urban development all around them. There's not many opportunities for gene flow, and the pollinators only travel maybe 1 or 2 kilometres. So I worked with some really fantastic geneticists. We found that they're clonal, but we actually didn't find high levels of inbreeding yet. This lower than expected inbreeding is great knowledge now, but the small effective population sizes, 3 of the populations having less than 10 unique plants, means that they will suffer negative impacts of inbreeding. And do need introduction of new genes if they want to remain healthy populations. Our last piece of missing information is germination. Fire certainly contributes, but in the lab, replicating these triggers hasn't been so simple. Working as a consultant, you are asked to move threatened plants because of developments in the way, and there is an assumption that if you move them, they will survive, and you don't have to maintain them, follow up and survey, see what's happened 10, 20, 30 years down the track. But I knew what happened 1 year down the track, and it wasn't great. Since that first failed translocation, it has taken us over 5 years and many thousands of hours to be able to confidently understand how we can conserve and translocate this species. This scenario plays out with many species around Australia and globally. More time and thought needs to be given before we default to translocation as a conservation action, because success takes a lot of work. Hello everybody, welcome back to session 2. Thank you so much, Christine, for springing that video on me. That's quite embarrassing. Now we have Dr. Tom May from Royal Botanic Gardens, Victoria. Tom, if you could jump on and get ready to share, that would be great. Tom's going to be explaining what socks have to do with mycorrhizal fungi and why these relationships are so important. And I would also like to give him a shout out and a round of applause, as I think Fungi Map is now a finalist in this year's Eureka Award. So well done there with that one. Tom, are you ready to go?
Okay, well, thanks very much, Chantelle, and thanks for the invitation. And look, it's great to actually be participating in an ANPC event. I've got a lot of very fond memories of editing Australasian plant conservation, and it's great to see a few familiar faces from a long way back. So I'm going to talk about mycorrhizal fungi, and look, apologies to those of you who are doing translocations. I don't have a lot of practical advice, but what I want to do is open up the topic for discussion, and probably more at the kind of strategic and policy end of things, but I'll try and pop some interesting facts in there as well. So first of all, I just want to draw attention to this fabulous web resource put together by Mark Brundrett about mycorrhizal associations. And there's also a great book that you can download called Working with Mycorrhizas in Forestry and Agriculture that has a lot of really good introductory information about mycorrhizas and the plants they associate with. So in simple terms, a mycorrhiza just means fungus plus root in ancient Greek. And so it's an association between green plants and fungi. And there are a number of different types, and they're formed by many different plants, and some of those types are restricted to certain plants, like there are orchid mycorrhizas, there are special mycorrhizas for ericoid plants. But two of the main types are the ectomycorrhiza, just meaning on the outside. So this is the sock. This is where the fungus forms a sheath around the very fine roots of a plant. So here, these incredibly fine threads, they're the fungal hyphae. They're about 10 microns in diameter. These are fine roots of a eucalyptus, and they're entirely surrounded by this sock of mycorrhizal fungi. So it's not some sort of accidental coming together. It's a definite structure, and it's formed routinely and repeatedly. The other kind of mycorrhiza, one of the major types, is the arbuscular mycorrhiza. And here we have a plant cell. So there's a microscope shot of a plant cell. And this is the fungus forming this wonderful arbuscle, which just means a little miniature tree. So this is a point of movement of nutrients between the fungus and the plant. Now, in general terms, most plants have mycorrhizas, and mycorrhizas have been shown to have positive effects on plant growth, both in the laboratory and in a field setting. So I've just picked two papers here. One is showing that if you get the right fungi out into the field when doing restoration, it improves outcomes. So this is an example, I think, from prairie grasses in the US. But there is a fair bit of controversy about exactly what's going on with each individual mycorrhizal interaction. Is it always beneficial for both the fungus and the plant? Sometimes one might be manipulating the other, and so on. So roughly we say that at least 80% of plants form mycorrhizae, but I guess there's just a lot more work to do. Actually, I'm just going to go back. I think I missed a slide. There we go. So a mycorrhiza is a symbiosis, and it's a particular type of symbiosis, which is a mutualism. So that means there's benefit to both partners. Now, fungi are heterotrophs, they don't fix carbon through photosynthesis. And so they gain sugars from the plant, and I guess there's a swapping there. The plants gain nutrients such as phosphorus and nitrogen. Those very fine fungal hyphae are really good at getting down into tiny cracks in the soil, and also fungi have a battery of extracellular enzymes that can mobilize and break down these nutrients into forms that are accessible to the plant. Mycorrhizas may also confer protection from disease, drought, etc. So I'll just flip forward to where I was before. So if we think about the millions and millions of trees and plants that are put out, both for just general restoration and for translocation of rare and threatened species, what about the fungi? And I would say vast majority of those plants that are put out, apart from orchids, no consideration at all of the fungi. We just sort of hope the right fungi are there, either in the soil that the plant's been propagated in, or in the soil where they've been put into, or somehow that the fungal spores blow in. And I think this is a really important thing to recognise, that we really just have no idea about that. And when you wander through a forest, I guess it kind of reflects the fact that we see the trees, but the fungi are hidden in the soil. The mycelium of the fungus, these very fine threads, it's invisible even if you pick up a clod of soil. We occasionally might see the sporing bodies, the mushrooms, the bracket fungi and so on that the mycelium puts up. But in effect, those fungi are hidden. So we don't really kind of notice them. And in fact, all ecology, I think, is biased in working on individual organisms. And there's a new concept called the holobiont, which is trying to emphasize that if you work on a tree, you have to work on all the things that are growing symbiotically with it. And look, I think this has come out really nicely with the orchids, with the pollinators and the mycorrhizal fungi, but it's really going to apply to anything. There are also numerous fungi that are endophytic within leaves, playing different roles in disease protection and so on. And then there are a whole lot of bacteria and archaea that live either inside those fungi or in other structures. Now, this is a diagram from some work done recently by Earl Bowd about the kind of cascading effects of manipulating a forest ecosystem. And the detail doesn't matter, but all of these lines are what happens when you do something. So in this case, Earl was testing the effect of salvage logging, not only on the trees, the eucalypts, the acacias, the shrubs and the tree ferns, but also on the different fungi in the soil through soil metabarcoding and at different levels. And it just shows that if you do something, it has effects, and there are indirect and direct effects. So the salvage logging increased the pH, that had a cascading effect onto the bacteria and so on. So don't worry about the detail, but what it's showing is that it's very complicated. And there's a lot of interactions going on there that we can't see and we don't understand. And I just think it's really important to start to try and get at all that, because some of these things are going to be very critical for plant health and persistence. Now, I just briefly want to touch on the fact that fungi are tractable organisms. I think for a long time people have thought it's all too difficult. What's the species of fungus? We can't see them. There's too many of them. They can be delimited taxonomically, and the species are just as real as in other biota. They can be readily identified. In fact, the DNA barcoding is really effective for fungi in a lot of cases. Many fungi can be grown in pure culture, and those that can't can often be grown in a co-propagation with a host, such as in the pot culture of arbuscular mycorrhizal fungi. And just one example for some recent work that's interrelations with plants is these Talisnella species associated with Australian terrestrial orchids. So this is work by Arild Arafin in Celeste Lynn's lab. And Arrow was able to delimit quite a number of new species. They all look virtually identical growing in culture. They all look pretty much exactly the same in terms of the hyphae. They don't have any sexual structures. but the DNA analysis across multiple genes very clearly delimited species. And whereas in the past we might have just said fungus isolate 1 or fungus species 8 or whatever, here we have a number of named fungi that have particular relationships with different plants. So in doing the work on the different orchids, it's important to be selecting the right fungus and knowing how to identify that. And that is all quite possible now. Now, just to touch on the one sort of thing I'd like to talk about in terms of practicalities, what I can say is that the source of inoculum for fungi is very challenging, and that's what I'll be talking about in the rest of the talk. So you can harvest spores from sporing bodies such as mushrooms through spore prints or from puffballs and use that as inoculum. I would say that moving soil is not advisable. It's not sustainable. It detracts from intact sites, and there's the risk of moving pathogens. Maybe at very small scale experiments, that's okay to take some soil from a good site and use that as the inoculum, but it's really not to be encouraged on a wider scale or as a long-term solution. Commercial inoculum is problematic. Often when commercial products have been examined, they do not contain viable spores. And if they do, the fungi in them may be exotic and potential weeds. So I would say work from adjacent intact areas in the sense that maybe putting your reintroductions next to a healthy forest that might have a good lot of fungi in it would be the good idea. But be aware that degraded sites may have altered nutrient levels that suppress mycorrhizal formation. So in effect, the plant says there's plenty of phosphorus and nitrogen here, I don't need a mycorrhiza, which may mean it's missing out on other benefits of the mycorrhizas like disease protection. There's a huge amount of research that needs to go on here. And I'm really heartened to be listening to the talks, seeing the translocation guidelines and seeing how much really good science is going on, answering questions about translocation and practices. And I think all of that kind of thinking and science, if it was applied to fungi, would yield some really interesting and practical results. So in terms of biobanking fungi, and I'm using the term biobanking because it's not just the spores. So a spore bank is a possible term equivalent to a seed bank, but spore banks actually, it's more than that because we may need to store the mycelium. It's quite practical, storing at -80 works. There are some appropriate preservation protocols that are needed, but it's possible. When you're dealing with fungi that cultures, they often look quite similar. It's really good to have quality controls, including sequencing to confirm identification. And a biobank for fungi would not only achieve conservation objectives in getting fungi themselves, just as we have seed banks for plants, conserve, but it's a source for research, such as on the taxonomy and the physiology. It's also a source for bioprospecting. A lot of really interesting compounds, including important antibiotics and so on, come from fungi. And it could act as a source for inoculation of plants if it's bulked up. And what I really want to emphasize here is don't be afraid of the F word. I think that over the years, I've noticed that when we start talking about the fact that there's 2 million species of fungi on the planet, everyone just goes, oh my God, they're going to take over. They're going to take over our research. There's too many. We can never get to know them. But I'd encourage people to embrace fungi and other microorganisms as integral components of holobionts, that is the organism plus all the symbionts, and of ecosystems, and recognise that you need to employ mycologists to handle fungi. They have expertise in isolating, culturing, preserving, in the taxonomy and identification. There might be special equipment required, but there are plenty of examples of labs out there that have it. And particularly, don't leave fungi till last. I've noticed over the last 4 decades I've been involved in fungal ecology and conservation that there's always a tendency, oh, we'll get around to the fungi. I will do some red listing of fungi when we finish the plants. We'll do a funga treatment, which is what we call a flora for fungi, once we've completed the flora. So I think it's much more important now to start working on fungi in tandem. Don't leave them till last. In terms of integrating fungi into biobanking, we really do need seed plus fungi banks. There is no point preserving all of the plants on the planet in seed banks if their associated mycorrhizal fungi are not also preserved. Because if worst case scenarios, if there's nothing left in the wild, those plants require those mycorrhizal partners. They might be able to germinate, but they won't necessarily thrive without those fungi. The resources are limited, but don't view it as a zero-sum game that somehow if you spend some of your limited pie on fungi, that will reduce the amount you can spend on the plants. I think a partnership approach is really important, and that we need to strengthen connections between outcomes for conservation, biodiversity in terms of inventory and taxonomy, and the other main area where we intersect with fungi, which is biosecurity around protecting borders and diagnostics. Building up a sound knowledge of our native fungi will benefit right across all those areas. And it may be that one way to do it is to build critical mass at some key institutions and then expand that outwards. One of the big issues that we face is that fungi culture collections around Australia are scattered and they're not integrated at all. There are fungal culture collections around the plant pathology area, obviously really important to do with plant pathogens. CSIRO holds a number of specialist collections that are not actively curated anymore. There are medical mycology culture collections with things like pathogenic yeasts, and then other niche areas like wine microorganisms. And there is no integration amongst these different institutions. There's no central place we can access the data, although the Atlas of Living Australia is helping. And unfortunately, historically, attempts to integrate microbial culture collections, this is bacteria, where you would think there is a really good use case. These are highly significant pathogens, they're used in industry and so on. Those attempts to integrate them and form some sort of national collection have been slow and yet to deliver, despite some really sterling efforts, particularly by Lindsay Sly, who has been writing about this for many decades. There was a short-lived ARC-funded network with an integrated culture collections database, distributed culture collections and so on, research networks. Didn't come to anything, and Lindsay's still working and writing about this. So we haven't been successful in getting a strong network of culture collections that are inside institutions where there's long-term protection for those cultures. And just as an example of what can be done, this is the CBS culture collection in Utrecht in the Netherlands. It's now called the Westerdijk Fungal Biodiversity Institute. They have more than 100,000 isolates. They have multiple -80 freezers with dedicated liquid nitrogen and so on. An incredible system. Everything is sequenced. And even there already, there is almost 1,500 isolates of fungi from Australia. So a lot of our fungi are being lodged there, and we're not really getting the benefit of having them lodged in a local institution. So just in conclusion, plants need fungi, and fungi need plants. And so I think a key role for ANPC would be to try and foster some sort of Australian fungal biobank network to get the fungi preserved so we can start to do the experimenting and the research to work out how to help you get better translocation outcomes. Thank you.
Thank you, Tom. That was great. I'm probably going to think of socks in a whole new way now when I put them on. There's a little question for you from me in there, but we're going to change tack now. Instead of plants, our next guest, Dr. Heidi Zimmer from CSIRO, is going to be talking about cash, particularly what is the cost and viability of translocations.
Hi, my name's Heidi Zimmer, and today I'm going to talk to you about the viability and cost of conservation translocation to manage threatened plants. I'm talking to you today about some work that I did when I worked for the New South Wales government, work within the Saving Our Species Science and Research Program. But today I work for the Centre for Australian National Biodiversity Research in Canberra, based at the Australian National Herbarium. Let's dive in. So, as I'm sure you already know, translocation is defined as population, establishment, and augmentation. So that means establishing new populations as well as adding individuals to existing populations. Conservation translocation, which will be the focus of this talk, is where conservation is the management aim. The aim is to improve the status of the species, reduce extinction risk, as opposed to mitigation translocation, which is the primary aim is to mitigate development impacts. So why would you translocate for conservation reasons? Well, species are affected by a higher extinction risk if they have a small or declining population size or small or declining geographic range. This is according to the IUCN categories and criteria. So Species with small populations and restricted ranges are at an increased risk of extinction, for example, from a single catastrophic event. It's like having all your eggs in one basket. So if you've got only one small population in one place and a big flood or fire or something comes through, the whole lot could be affected. Whereas if you have more individuals in more populations, then there's less chance of a catastrophic outcome. And translocation is useful because you can add individuals to your population, but probably more importantly, you could make a new population somewhere else where it's not going to be affected by the same threat. So, translocation in the New South Wales Saving Our Species program. Translocation was surprisingly listed as a management action for 30% of site managed plants. That's plants that are managed at a site-based scale, and 51% of critically endangered plants in the site managed program. Now these are really big numbers because worldwide translocation was only listed as an action for 4% of threatened plant species on the IUCN Red List, or 12% of critically endangered plant species. So in Australia already we're doing translocations at a much higher rate than globally. And here, yeah, you can see that about half of critically endangered species had it listed as a management action, a third of endangered species, and about a quarter of vulnerable species in the program. So why is this so? Why are people picking up translocation in New South Wales so much? Well, 45% of these site managed plant species were being listed as affected by demographic threats. What is a demographic threat? Well, when you dive into it, it's usually the threats associated with having a small population size, like demographic stochasticity, like inbreeding or environmental stochasticity. So it's like I mentioned before, having a disturbance take out the whole population. Interestingly, 88% of species with translocation as a management action were also listed as having demographic threats. So it's being identified as an option for these species with small populations and limited distributions that are at higher risk of extinction because of this. So translocation for 30% of site managed plants, but isn't that expensive and risky? Historically, translocation has been thought of something that is expensive and risky. So let's take a look at that. Using data from the Saving Our Species database, it was a couple of years ago now, and these were estimates by people who were planning to undertake these projects. I looked at the costs of management, and I found that compared to other management actions, such as fire-related management actions, pest, animal, and weed related management actions, translocation management actions were actually pretty similar, in the order of about 100,000 but per species, but hugely variable, of course, and we'll talk about that a bit in a second. So yeah, is translocation more expensive? Well, not really. Like in terms of estimated costs, it was pretty similar to pest animal control and fire suppression. So is it expensive? It depends on your frame of reference. And is translocation risky? I'm sort of asking around and looking at things, why people thought translocation might be risky. I got the feeling it's because it's seen as being quite active or quite interventionist. And maybe there was a perception that the upfront costs were high, which they are in propagation of the plants, and establishment of the translocation site does mean high upfront costs. And I will note here that at the time of this research, the Royal Botanic Gardens and Domain Trust in Sydney did have a framework for estimating costs. And of course, growing things such as orchids, which have complex germination requirements, were much more expensive than growing things that were simpler to grow, like an acacia. So this comes into the high variability, but overall, the upfront costs were high in terms of growing the plants, establishing the sites, and then with generally lower costs through time with site maintenance if the sites are established well. And people might have also thought that translocation was risky because they'd had limited experience with successful translocation. Of course, through fora such as this one, people are learning about more successful translocation experiences, and of course with the wonderful updated guidelines, people can see plenty of examples of successful translocation. So the other side to talking about cost is talking about benefit. And what is the benefit of translocation? Well, we're hoping that, well, it would be the aim that the translocation would increase the species viability in the wild. And what does that actually mean? It's the difference in probability that the species will persist with and without the management action. So going back to our example, or the saying, all your eggs in one basket, if you, for example, establish a new site and at a new location which isn't affected by the same threats, you're halving your extinction risk. And so translocation can have quite a significant benefit. So if you're not sure whether your translocation is going to benefit your species, you've got to remember that if you've got a small population size or a small geographic range or declines in either of those, your extinction risk is increasing. And so translocation can be used to mitigate one or both of these. I'll talk you through some examples. We made a decision framework as part of this study. So the first example is where the benefit from translocation is likely to be high. These are when the populations are very small, like critically endangered, with fewer than 250 individuals and usually existing only at one site, so a very restricted geographic distribution. The species is therefore threatened by a catastrophic event like the one that, like a fire or a flood or a disease. One bad thing happening could extirpate that species from the wild. The outcome of a successful translocation would be spreading the species across more sites with a larger population, thereby decreasing the risk of one bad thing happening. The example here is, the population is so small that for example, a small impact could basically affect the whole lot. Whereas if you made a second population somewhere where that impact isn't occurring, you're halving your extinction risk. In the next example that we have is the population, the species doesn't necessarily have to have a small population size, but it has a threat which you can't manage at the site. And a good example of this might be Phytophthora. So it's really hard to get on top of, and the species is declining, and you can't manage it so that to arrest decline. So mortality is occurring at a greater rate than recruitment. So the threat's ongoing and you can't manage it. Here you could create a new population at a new site which isn't affected by that threat. Now here the benefit though is only moderate, because really one of the aims of translocation is that you keep the wild site going well. So you've got to have two happy functional sites to reduce the extinction risk, not just one. And so here the benefit is moderate because of course you can't get on top of the threat at the wild site, but you have had some benefit in creating a new population at a new site where the threat isn't present, hopefully, through good recipient site selection. Here's a third example where translocation is likely to be less beneficial. Here we've got a species which isn't critically endangered. It's not particularly restricted, doesn't have a particularly small population size, and it's not declining. It's pretty stable, but it's not increasing either. So would you do it? I'm not sure. There are likely to be much higher priority species among your critically endangered ones, or ones that are declining. You might think about it if you really wanted to increase it, like if it was reproductively limited, but there's likely to be much higher priority species. And in a fourth example, we have a species which isn't critically endangered, doesn't have a small or restricted population size, and it's not declining. You're able to manage the threats at the site so that it is stable, at least, if not improving. And again, it's going to take a lot of translocating and a lot of new sites and new individuals to have a big impact on the extinction risk. And so, yeah, it's really unlikely to be cost effective if it's possible just to manage the threats at the site or the site's going along just fine as it is. So yeah, there are likely to be much higher priority species like the critically endangered ones we talked about at the top. And the last example we have is mitigation translocation, and really in terms of extinction risk theory, moving your population out of the way of a development from one place to another isn't really reducing the risk of species extinction overall, because you've got the same number of plants still at one site, so not much benefit there. So some key points here. For translocation to have a big benefit, your wild site must be maintained. So you've got to keep that original site going. You can't just swap one for another. You'd still have all your eggs in one basket. So it's important to make sure you're on top of management of the wild site as much as you can be. And it's really important to be clear on what the most important threat is. So that helps with wild site management, but it's also really going to help you with recipient site selection. You want the recipient site to be free of the threat that was affecting the wild site. And of course, it's a oldie but a goodie and a bit of a hard one, but your translocation has to be successful. And success is defined as the translocated individuals producing viable offspring and those offspring producing offspring. So this is a long-term proposition. Translocation isn't a quick fix or even a cheap fix, but if you think about the benefits in terms of reducing extinction risk, it's definitely worth it in some cases. So is translocation expensive given the potential benefit? Well, you've got to assess that on a species by species basis. And it is possible to reduce the costs by following best practice guidelines and learning from others. So following best practice guidelines, you can, for example, minimize the mortality of the translocated plants, and you can plan to maximize your extinction risk reduction by putting your recipient sites in areas where the threats aren't operating or at least reduced, and you can manage to avoid negative impacts on the recipient site. Obviously, as has been discussed in this forum, monitoring is important. We found in the review of the data that monitoring was recommended for 99% of plant species, which is really good in the SOS program, but it's particularly important, of course, in translocation situations so that you can head off any problems before they happen, and also make sure that you're tracking for translocation success. And it's important to note that obviously, because of this symposium, our understanding of translocation is evolving rapidly. So it is possible to follow best practice guidelines, learn from others' mistakes, and avoid unintended consequences by keeping abreast of what's happening and doing your best to follow the best practice guidelines. And it is possible also, if there are any management questions or ecological questions, to build those into monitoring design. And this can help address uncertainties of translocations going forward and increase future success rates. So what are the key messages? Translocation is most likely to reduce extinction risk in species that have very small population sizes or very restricted distributions. And you're doing this by ensuring all your eggs aren't in one basket. I'd like to thank my funders, our funders, which is the Saving Our Species Science and Research Program, and the co-authors on the paper that was published in Australian Journal of Botany back in 2019, Tony Auld, Peter Cuneo, Kath Offord, and Lucy Commander. And thank you very much for listening.
Great. Thank you, Heidi. And thanks everyone for bearing with us. That's excellent. There's some questions in the chat for you. Warren is joining us again. Warren, you can pop on screen if you like, because I've done my introduction. Warren joined us last week to talk about propagation methods, and he's now going to be talking about the role of living collections, hopefully with some more fun examples from all those years of experience. So Warren, please take.
Change the title just a little bit from role to roles of living collections, because there are a lot more than just a single role for any living collection. But before you start looking at the roles, need to be looking at doing background work and conservation or preservation. I like a lot of the legumes, so I've decided to use a lot of peas in the topics that I'll be covering. And I'll go through each of these: the purpose, planning, partnerships, preparation, prosecution, and perpetuity of ex situ material collections. So at the start, make sure you have a purpose for the collections you're doing. Justify the needs and the roles of what the collection will be, the how, when, where, why, etc. of will the collection be used. And for a start, one of the roles might be the living collection. It's just a display used for public education and horticultural education or botanical education. Might be the regeneration of collections which are already in cultivation in an ex situ location. It might be to support the translocation, as Heidi was just talking about and others have spoken about this week and last week. It could be the core of an ex situ collection, and this is becoming more popular with climate threats, climate change threats occurring in a lot of natural habitats. Ex situ collections are being established to where they can be managed more closely than translocating to natural habitats where they're out of view of regular monitoring. It could be the core of a reintroduction collection. So it may be that you want to actually build up the numbers of plant material that you're going to use for reintroduction. So that could be by seed production or vegetative propagation purposes. You might be wanting to establish just a seed production site or a seed orchard to build up your seed supplies of any threatened taxa. It could be field gene banking, where the field, the genetic diversity is quite limited, and you may need to ensure that the genetic diversity is preserved of the taxon you're working with. It could be research for students, and it could be the staff within your organisation or outside the organisation. The research in particular, RBGV Cranbourne, for instance, we get quite a number of students from universities in particular trying to source plant material that is extremely rare in cultivation and may only be available to get material from them by going to wild provenance material. So the ex situ collections helps actually prevent or minimise the impacts of researchers going out into the field. And distribution of germplasm for other conservation organisations. It could be sharing plant material as a meta collection, or it could be conservation work that's being done by other institutes. The planning part of it, I mentioned last week, and it's an absolute essential, as everybody I think who's given a talk so far has emphasised. Your planning is essential, getting your right permits, whether it's private landholders, state, federal. Accurate records, so look up and research where you're actually going to get your material from, and research and record where you've actually collected the material from. And it's important to have that accurate information for reintroduction purposes. Herbarium vouchers are essential to validate the actual collection that you made, just to ensure that you've actually collected the right specimens, or to have qualified botanists to validate that information. Long-term management, think about what you're going to do with the plant material once you get it back in and growing successfully. Have you got long-term goals planned to actually utilise the material effectively? The nursery resources in the short term, do you have the infrastructure and the equipment, the skills, biosecurity, quarantine, if you've got sensitive plant material, to actually grow them successfully in a nursery and ensure that you can preserve them. Ensure the participation of experienced horticulturalists to help validate all the work that you've been doing. So specialised conditions may be required in the nursery. That might be aquatic plants, they might be alpine plants, plants that grow in very specialised soil conditions or substrates. So all these sorts of things need to be researched before you go out in the field or even consider going out in the field. Are the plants going to remain in the nursery ad infinitum, or are they going to be planted ex situ or used for translocation? So even when you get to the stage of hardening material off or wanting to use them for translocation, make sure that the plants are in a condition that they can be going into the field in a similar condition to which the outside conditions. Don't take them straight out of a glass house, straight into subalpine conditions, for instance. Planting out, make sure your site preparation is done well in advance. The planting time is done correctly. Follow-up management. Earlier on, we saw just small fencing around planting sites. It's the sort of thing that protection of plant material in advance prior to planting is well and truly demonstrated. And material handling, storage and transport of logistics. There's all important things to consider. Classic example is the eastern bristlebird movement from New South Wales down to Wilson's Promontory, just done recently. All sorts of loops had to be jumped, or hoops had to be jumped through to actually achieve that successfully. Partnerships are really important. It's already been emphasised a few times. So one of the examples that has been promoted in the germplasm guidelines in Chapter 8, Joe McAuliffe has a private property in bushland outside of the Canberra territory, and Joe's property ended up having a number of plants of the Grevillea Renwickii, sorry. So from his plants on his site and nearby, 16 samples were taken from different plants, and the genetic research that was done on that identified that there are only 2 separate genotypes there. So it was possible to de-accession quite a few of those plants and just take it down to 2 accessions. From a conservation point of view, this is really important because it would imply that potentially you only need to have basically 1/8 of the actual material in cultivation in your nursery or being planted out if you're doing ex situ planting in the open ground, to ensure that you actually maintain those genotypes. So working with geneticists from an early stage, in this case, has been extremely valuable. Another partnership, there's one that I've partaken in with the Australian Tropical Herbarium, James Cook University, and all the participants listed here. So quite a big project and quite a few different diverse participants in it. And the project was aimed at conserving plant material from the high mountain peaks of tropical far north Queensland, so in behind Cairns. Part of what Tom was talking about with the plant material was that the fungi collections by the Australian Tropical Herbarium, they only had 7 fungus species collected from this area, that's Mount Ballon and Kerr, in their whole of the collection over many years. Collections have been made by botanists up on the top of the mountains, but only 7 fungi have been collected. And if you well and truly know the rainforests of far north Queensland, there are far more many fungi species growing So we, over and above collecting the threatened higher plants that were recognised as being requiring collection, it was important for us to collect for the herbarium and collect the fungi as well. On different peak, we ended up collecting the Eucryphia wilkii. So the bringing of Eucryphia wilkii from the top of Mount Bartle Frere, the highest peak in Queensland, and reintroduction into the Botanic Gardens at Cranbourne, where plants are growing successfully in a couple of different locations. And these flowering plants have been growing successfully at the gardens and flowering for about 8 months continuously now. When we were collecting from the top of Bartle Frere, trying to collect seed from the diversity of plants that we could find, which was only 5 plants across, which we had found after about 2 hours of searching, we only collected about a dozen or so seed heads. So by bringing these plants back into cultivation, it's giving us the opportunity to collect further seed. And at the moment, there is the genetic research being undertaken to find out whether the individual accessions we have are all the one clone or whether there's a range in genetic diversity. So the meta collection of this material that we collect at the high mountain peaks, there's plant material being grown at the ANPC, the Dandenong Ranges Botanic Gardens, and the Royal Botanic Gardens Cranbourne, and Melbourne Botanic Gardens. And there are other institutes who have actually taken plant material from these collections, so going to South Australia and back up to Sydney Botanic Gardens. And an expression of interest from Tasmania, and we'll be sending material down to Tasmania as well, all in the hopes of making sure that the diversity of the genetic material we've collected is being preserved. So the preparation, if you have got all of the previous items, the planning and so on out, if you've done those correctly, then you'll get to the 3 rights for preparation. You'll have the right resources, you'll get the right plants, and you'll be out in the field at the right time. So prosecution, the same situation. You'll actually be out in the field at the right time with all your resources that you need for effectively collecting the diversity of plant material that you may be introducing back into cultivation and or trying to save. You're making sure you've got the plant identification correct, and timing for collection of cuttings, etc. is important. So not in the middle of summer, etc. And if you've got all of these rights correct, then you'll get down to the perpetuity, the collection of the material and the plantings. You have the right plant in the ground, growing at the right time, and you'll end up with the right results of preservation of the material. And I should say at this stage, hopefully I haven't jumped into what Amos was proposing to talk about. I understand from Chantelle at the start that Amos was going to be talking about rights. So we'll find out whether there's a clash or not. Thank you for that. And it looks like I've actually finished a bit earlier too.
Thank you, Warren. Very punctual. I like those 3 rights, very clever. And I also think the role of meta collections is going to become increasingly important. Now, Dr. Mark Freestone is joining us all the way from Spain. Thanks, Mark, for being online. But we are going to play a pre-record because he has unstable internet at the moment, but please ask him any questions in the chat because he's up and happy to chat.
Good afternoon, everyone. My name is Mark Freestone, and I'm currently doing a bit of contract work for the federal government, trying to get a few more plant species listed on the EPBC Act. But I'll be talking this afternoon about my PhD project, which finished a little while ago now, but was looking at these wonderful orchids called leek orchids and trying to stop them going extinct. I was based most of the time at the Royal Botanic Gardens Victoria with Mushka Ryder, and also part of the time up at ANU with Celeste Lind, and Nigel Swartz at UTAS was also a supervisor. And apologies, this is a pre-record because I'm currently living over in rural Catalonia in Spain, and yeah, probably enjoying a sleep in right now. No, I'm sure I'm not. I'm sure I'm watching. Prazophilum can seem like a kind of a niche study species, but to talk them up, they're the third largest genus of orchids in Australia, so about 140-ish species in total, 50, 60 species in Victoria, depending on your taxonomy. And their centre of diversity seems to be fertile soils in the southeast of the country. So that's where a lot of their sort of habitats that have experienced a lot of agricultural impacts. And so it's no surprise that there's a lot of threatened leek orchids. We've already lost a couple of species to extinction probably. And Prazophilum is the 5th largest genus of all plants on the EPBC Act by number of species listed. So yeah, there's a lot of threatened leek orchids. And that's not a problem unfortunately, that's not a phenomenon that's particularly unique to leek orchids. There's a lot of endangered plants in the world, but in Australia. But leek orchids have got this kicker that they just weren't able to be grown reliably in cultivation. For some reason, the seed wouldn't germinate. And so a quick refresher on orchid propagation methods. Because orchids have tiny seed, they need mycorrhizal fungi. So to grow them in cultivation, you need to go out and collect some seed and some fungi from the roots of a wild adult plant, bring them back to the lab, grow the fungus up on a petri dish and sprinkle on the seed. And unfortunately, most of the time, this is what we got when we tried to grow leek orchids, which is not much. And that's a problem if your Prazophilum gilgai grows in a tiny little reserve up near Benalla, last seen in 2016, and myself and a local naturalist up that way, Sally Mann, have been looking for it every year since. And there's been a few wet years in there and we haven't found it, and it's really concerning. It could be the most recent Victorian plant species to go extinct. Hopefully that's not the case, but it's certainly very close. And it's followed by a huge wave of many really highly threatened Prazophilum species. Correctum from central Gippsland, only known from a couple of plants. Its population has crashed over the last 15-20 years. Prazophilum fosterized, only known from a single roadside grassland out past Geelong. And reedii I put in here, not because it's got a particularly tiny population, although there's not many plants, but it's only known from a single swamp on private land that's not protected in any way, shape or form. And it's a really vulnerable site. I could only fit 4 species kind of nicely on this slide, but in reality there's heaps of highly threatened leek orchid species. I could probably spend the whole presentation talking about them all. They're in a lot of trouble. They face threats to their wild populations that are difficult or impossible to manage, and their wild populations for many species will probably go extinct in the near future. Unless we can get them into cultivation and implement the sort of breeding and reintroduction programs that people like Nushka have done so successfully for a whole heap of other similarly threatened orchids. But we can't do that if we can't grow them. So my PhD was working out how to grow leek orchids. We had a few theories, not short on theories. The first one was seed viability. Some investigations showed that it could be a bit low in some, or probably is quite low in some leek orchid species, but it's not low enough to explain the lack of germination across the board. So, and it was kind of a boring chapter in my thesis, so I'll gloss over that one today. Much more interesting is the role of mycorrhizal fungi and the impact of the nutrient conditions in the germination media that we're growing these things on in the petri dishes in the lab. But first to the fungi. So leek orchids, like all orchids, have tiny seed. Seed so small it can't germinate by itself. It just falls to the ground, and rummaging through the soil are these little hyphae of these mycorrhizal fungi. And for some reason, when they stumble upon the seed of a leek orchid, they don't eat that seed, but they grow into it and pump food and water into that seed to get it to germinate. Why they do that, we have almost no idea. It's kind of one of the beautiful, beautiful sort of unknown phenomena of the world of botany and mycology. Yeah, that they do, and that's how the orchids germinate. We couldn't get this to happen in the lab though, so we thought that there must be something going on in this interaction between plant and fungus that's causing these problems in the lab. So first of all, I did a bit of a deep dive into the world of the fungi and the roots of adult plants, because it's from the roots of adult plants that we extract the fungi that we use in the germination trials in the lab. So what's going on in the adults? My first study species was Prazophilum lindleyanum from up on the granite outcrops in the Warby Ranges. And I sampled the roots of adult plants at 4 times in a growing season. So just after they emerged in autumn, in early winter, late winter and spring, and sequenced the fungi that were living inside those roots using DNA testing. That's how we identify these mycorrhizal fungi. And I found 7 species of fungus growing in the roots of the adult plants, these represented by these orange vertical lines here. So 2 species were really common, really frequently detected all throughout the growing season, and 5 species were found only occasionally, which is interesting because leek orchids have a kind of, it's not well known by the general public, but Australian orchids, have a reputation of having quite specific mycorrhizal fungi associations. So lots of species, Calodanias, Diurus, Pterostylis, only associate with 1 or maybe 2 species of fungi. But here we have an orchid associating with 7 species of fungi, which is a little bit unusual. We couldn't help but wonder which of these fungi species was involved in the germination process. To work that out, we made up some tea bags, glorified tea bags really, filled them with orchid seed and buried them around the base of the wild plants in autumn. Watered them every couple of weeks because it never rains when you're doing a seed baiting study, and had great help with that from the city of Wangaratta. And then dug them up in spring. And you can see here wonderful germinating leek orchid seedlings and protocorms, these sort of germinating seeds. And on the microscopic photo on the right, you can see these little white or rusty orange coloured sort of cotton wool ball type things. They're the coils of mycorrhizal fungi hyphae, like the little roots of the fungus inside the protocorm that are feeding that protocorm food and water probably to get it to germinate. It's a beautiful, beautiful thing. So I grabbed my scalpel and cold-heartedly cut up all these innocent little seedlings into tiny little pieces and sequenced the fungi, and found that every single seedling and protocorm only associated with 1 of the 7 fungi that are found in the roots of the adult plants, which was interesting, hey? Because maybe this could explain why we're having such troubles, because maybe we've been sampling the wrong species of fungi from the adult roots. Anyway, that theory started to fall down pretty quickly because I looked at a couple of other study species. This is Prazophilum validum from out at Deeplead near Stawell. The adult plants also associate with 7 species of fungi, but so do the germinating seedlings. And the most common, most abundant, or frequently detected fungus also was involved in germination. So it seemed a little bit unlikely that just randomly missing this common abundant fungus was going to explain our problems with germination in the lab. And the theory totally fell apart with Prazophilum frenchii, which does have a very specific association in the roots of adult plants. Adults only associated with a single species of fungus, which was also involved in the germination process. So there was really no clear evidence that sampling the wrong fungi was responsible for poor germination. Finally, I'd like to talk about the role of the nutrient composition of germination media. A little bit more agricultural, I guess, and less, a bit less scientific, but actually quite important. So normally when we try and grow native orchids with their fungi like this, we use this sort of porridge-based mixture. This is the jelly sort of stuff in the Petri dish. And that seems to work pretty well for calladenias and pterostylis and Dwyer and all sorts of things. But it never, people never really had much luck growing leek orchids. So I did a big symbiotic germination media trial, and first of all looked at the role of this sort of industry standard oatmeal agar mixture, if you can call growing endangered orchids an industry, and found that, confirming our previous observations, this oatmeal agar is pretty useless for growing leek orchids. Didn't germinate anything. But when we added in a few more nutrients, hey, we got a bit of germination. Not huge percentages, but reliable germination when we added more macronutrients or changed the carbon source from oatmeal to wheat bran. So that's great, fantastic. We're very, very happy with that. Interestingly, though, this is Prazophilum frenchii. Remember, the adult plants only associate with a single mycorrhizal fungus. I tested 3 isolates of that fungus, collected quite far apart, so we're pretty sure there are 3 different individuals of that fungus. And these 3 different individuals of fungus prefer different nutrient media. This one this green one on the right hand side likes the extra macronutrient mom, but only if it doesn't have sucrose. If it's got sucrose, like in the box to the left, if just normal mom, it hardly germinated anything at all. And similarly, the red one on the left only germinated seeds when it was on a very nutrient poor wheat bran medium. Hated the high macronutrient mom media. So these 3 individual fungi were from the same species of mycorrhizal fungus, and they had almost identical DNA sequences. So there's considerable diversity among individuals of mycorrhizal fungi within a species of mycorrhizal fungus, and maybe that can explain why orchids can occupy, slightly different micro habitats or little niches, slightly wetter or drier spots, or because these mycorrhizal fungi infer or confer some type of ability to change a little bit, to adapt to their environment. And if you're doing a translocation project, it's possibly quite important to retain this diversity in your mycorrhizal fungi that you've used to grow your orchids with. Anyway, I'm getting a bit off track here. We grew some leek orchids, and that was great. Germination rates of about 5%, but orchids produce so many seeds that the 5% germination rate is actually pretty good. And they grew really well and vigorously in the nursery and started to flower about 18 months after they were sown, which was very vigorous. So it turns out that the nutrient composition of symbiotic germination media is really critical, and not just for Prazophilum frenchii, but for a whole heap of leek orchids. We had a really good germination event with Prazophilum fosteri and Prazophilum correctum, and I think I germinated almost 20 species of leek orchids, and since I left, Nushka's probably pushed that up a fair bit more since. So that was great. That was kind of the key thing that we were getting wrong. It's interesting why leek orchids need this different media. I don't really know. I suspect it's probably something to do with them growing naturally on relatively fertile soils, and so maybe the mycorrhizal fungi have more or require more nutrients in their soil in order to have enough extra food to give to the germinating orchid seeds. Who knows? But yeah, for whatever reason, it seemed to work. And now the nurseries in the Botanic Gardens at Cranbourne are full of some really important insurance populations of some very endangered leek orchids, and seed can be collected from those plants, and they can act as a springboard to for future larger scale propagation and reintroduction programs. To finish, I'd just like to touch on the broader implications for orchid conservation. Again, getting back to that point of maximising the diversity of mycorrhizal fungal isolates. This is Prazophilum lianum from the Warby Ranges. Again, we met earlier. Adults associate with 7 species of fungi, but only 1 is involved in the germination process in the wild. Using our newfound media, I was able to test these other species of fungi to see if they germinate the seed in the lab. And concerningly, they did. Every species that was tested germinated the seed in the lab just fine. And that's a little bit of a worry, because most of the time when you're doing a propagation and reintroduction program for a threatened orchid, you don't have all this information on the seasonal associations of the adult plants or the seed baiting results, all of that. All you have is which fungi you've sampled from the wild germinate the seed of the orchid. And it's really important to maximise the diversity or number of isolates or samples of fungi that you use in that when you're growing plants for reintroduction, because if you don't, there's always a chance that you could be missing a fungal species that is really important in the life cycle of that orchid. So using a diversity of mycorrhizal fungi is likely important for the success of reintroductions. And I guess finally, to touch on some possible broader implications for other non-orchid plant translocations. Orchids tend to get tend to be quite popular and well studied relative to other plants in general, perhaps. But particularly when it comes to researching their relationships with mycorrhizal fungi. But we know that actually 90% of all plants have associations with mycorrhizal fungi, or others have associations with symbiotic bacteria, and they associate with these fungi and bacteria for a reason, right? Because they might help the plants to survive periods of environmental stress like droughts or floods, or give the plants some competitive advantage that enables them to hold their own, in the environment, or confer some disease resistance or something like that. But we know almost nothing really about the role of mycorrhizal fungi and bacteria, symbiotic bacteria, in the success or failure of plant translocations. And it's kind of a bit harder, I think, than dealing with orchids, because with orchids, they're so extreme, like they won't germinate without their fungi. So it's a really obvious problem that helps you to build quite makes it easier to know what things to research, eh? If you can't germinate the orchid, you need to work out why. It's a bit harder, I think, when the problems are not so well known, and designing an effective research project is a little bit more tricky, I think, for some other plants. But I think it's probably quite likely that that the role of mycorrhizal fungi and bacteria is quite important in the successful failure of plant translocations, and to work that out, we need more research. Big thanks. This was a big project with heaps of people that contributed really valuable information and time. So big thanks to everyone who helped me, particularly to the Herman Slade Foundation, the former NESPUB, and DELP, who provided the funding for this project. And I'd like to acknowledge the traditional owners whose land I undertook field work on, and who've been looking after these leek orchids since time began. So yeah, thank you for listening.
Thanks, Mark, and thanks for interrupting your sleep in. That was really thorough and also probably illustrates the importance of deep diving before arriving at conclusions or semi-conclusions in your case. Thank you to all the presenters that were involved in this session. We are going to have a break now and come back at 3:35, where we're going to be talking urban mistletoe, right way science, Goldilocks, and the urban frontier to finish up. If you have any questions, pop them in the chat or the Q&A. Q&A is working for some people, but not others. I would encourage you to do that. Okay, have a nice break.
This is Pimelia spinescens, or the spiny rice flower. Occurring on the volcanic plains of Greater Melbourne, this vulnerable species has been moved more for developments in the past 23 years than any other species we know of. So you would think, after all this time, successfully relocating plants so that they survive and recruit would be a refined art. But as this story will show, success doesn't come easily, and someone needs to be checking.
Rather than what was normal for the day was once an area was designated for development, the environment just got bulldozed. And we just come up with the idea of, well, how about we pick these things up and move them into the reserves. When we found a tree spade, which basically hydraulically pushes spades into the ground and pulls out a cone of soil, and you do that in the place where you want to put the plant, and then you throw that lump of dirt away, and then you get your plant, and then you plonk it into that hole, and then give it heaps of water.
The salvage method Steve developed has become an industry standard. The issue is, no one knew how many translocations there had been, and if they even worked.
Picking up a plant and moving it from one place to another is one thing. It's trying to put it into an environment which you then manage as a natural environment and try to get a population established.
According to Australian law, developers could relocate a plant in the way of a development and were only required to maintain, monitor and report on its survival for the life of the project, historically 2 years. In the context of a more than 50-year lived species, 2 years is not enough. These short time frames meant that opportunities to learn and improve methods were limited. But luckily for Pimelia, someone else was working on this species.
I planted that plant. That is a translocated plant that I grew from seed, and it is alive. Oh, I thought it was dead actually. Oh, that is so exciting. I started working on this species when I started my PhD, but I certainly met this species, I think, when I moved into the area. And I went for a walk and I found the species. I found a book, found out what it was an endangered species. I decided to go back to uni and study the environmental science.
Steve and Debbie are both part of a recovery team for this species, a group that advises on how best to conserve it. But without species knowledge, advice is hard to give.
Babies!
Debbie's research about its ecology has been crucial for understanding how to propagate this species, enabling the replacement of individuals lost through development. She was also the first person to successfully grow it from seed and has been perfecting the method for over a decade.
This is seed that I've collected since 2009 probably, and we're still reusing it to grow plants for people now.
But that wasn't the only thing Debbie and the team found out. The 2-year monitoring reports we talked about earlier were often private and notoriously difficult to track down. This limited opportunities to learn what worked and what didn't.
A lot of the reports are written for developers, they're owned by developers, but I think there should be perhaps more public documents about what's going on.
Lack of transparency is a problem in many mitigation translocations. But in this case, the team decided to do something about it. They have stipulated that 50% of plants must survive, and if they don't, they need to be replaced.
We put in the management plans in that once they go below that, they have to replace them.
They have also stipulated that reports, including successes and failures, should be sent to them so that everyone can learn what has and hasn't worked. And they have set a minimum management and reporting period of 10 years. One of the projects that requires these rules to be in place is managed by Brimbank City Council.
So we're at Pioneer Park Grassland in Sydenham. So this section in particular is quite a good C3 dominated section of grassland, of which we don't have that much in our reserve. So a few different species that we'll find in here, so Ostrastipa bigeniculata, Ostrastipa curticoma, and also some Ostrastipa gibbosa in here. More importantly, in here, we've got translocated Pymelia spinescens. They are surviving currently at about 38% of what went in. Because the translocation has fallen below 50% survival, we basically want to put more genetics in and more individuals in and keep those numbers up and hopefully get them to pollinate.
The other thing Brimbank has done, which is critical to the recruitment of Pymelia spinescens and the health of the grassland as a whole, is reintroduce a 2 to 3 year fire program.
This is the plant recovering after its burn 3 weeks ago at least now. You can see all the new growth on it. So they need fire to be healthy and to promote germination of seed and to open up spaces. It's vitally important.
Once you get to the stage of having a frequently burnt grassland, trickling is a really good way of describing the fire behaviour. So yeah, it just trickles and you don't really put any heat into it and it just kind of meanders along.
With developments only set to increase around Australia, mitigation translocations will become more frequent. But the vast majority of mitigation translocations still only require monitoring for the life of the project, and sometimes less, which is not enough. The Pymelia spinescens story represents one of the best examples of mitigation in the country.
You think global, act local. You have to look after what's right under your feet. It's so important.
We're learning things that otherwise we would never have learnt. Even the Commonwealth have looked at us as a potential model for setting up recovery teams.
And it is still far from perfect.
There's 3 plants left here that we know of, one there and two over there, and that's it in this whole reserve.
Well, what are we now? We're 20 years after the fact, and about half of the plants are still alive. I suppose these salvage translocations are as much about learning about the species and learning about the environment that they live in so that you can make better decisions later on.
Maybe the future of conservation, particularly in urban areas, will have to rely on more collaboration with developers, showing people what a well-integrated urban natural landscape could look like. But currently, we are a long way from that reality.
Okay, so our final session, we have 4 presenters, all of which I'm pretty intrigued about. Urban mistletoe, Goldilocks, Urban Frontier and Right Way Science. In our next session, Professor David Watson from Charles Sturt University is going to be talking about what's maybe a hard sell, but maybe I'm wrong, the role of mistletoe and urban introductions, if that's right. All right, take it away.
Thanks, Chantelle. Lovely to be here. I'd like to acknowledge that I'm coming to you from Wiradjuri country, and the work I'll be presenting was undertaken in Melbourne on Wurundjeri Woiwurrung and Bunurong Boonwurrung peoples of the Eastern Kulin. So they're the traditional owners of the land where this work was undertaken. So mistletoes in street trees. What's that all about? So I'm going to start by giving you a bit of an overview of mistletoes, what they are in the plant world. They're kind of unloved, as Sean sort of hinted at, and then walk you through some of the main interactions that we know about that involve mistletoes, which then gives you a reason why you might want to put them back into an urban forest where ecological interactions might be missing. Then I'll review what we did, what we found with this inoculation study, and then point to where we're going with this, both this particular study and broader work on mistletoes and their role in a variety of landscapes around the place. So let's get to here. Oh, no, that's the wrong thing to do. Let's see if I can do, there we go. Okay, mistletoes. So mistletoe is a functional group. It's a way of being a plant, like a succulent or a mangrove. A mistletoe is an aerial, woody hemiparasite. And that's a way of being a plant that evolved 5 times independently, all in the sandalales and all from root parasitic ancestors. So 5 families of flowering plants are not each other's closest relatives. So they're a polyphyletic group. And there's 3 of those families represented in Australia. The one that will be most familiar to most of you is the Loranthaceae, the showy mistletoes with the big bright colourful flowers. Australia has 2 of the 3 weird root parasitic species of Loranthaceous mistletoe, including the West Australian Christmas tree, which has woody winged seeds. Everything about that plant is bizarre. But we're talking about Loranthaceous mistletoes, the typical ones you'd see as you're driving around. And please don't think that we know everything we need to know about these plants. So this species here with the long tubular flowers, that's an unnamed species of Psittacanthus that I found in Western Colombia a couple of years ago. And then doing field work just south of Meekatharra 2 years ago, there's this Lysiana that popped up that keyed out as unknown. So there's still a few mistletoes that we're trying to work out who they are. In Australia, there's 97 species we know of. We're probably going to get to 100 species pretty soon. With this audience, I don't think I need to specify, but most people, including many people I talk to at ecological conferences, are surprised to learn that they are native plants. So this critter here, this succulent thing, some of you might recognize that as that is Viscum album. That is the quintessential mistletoe in the Viscaceae from Western Europe, native to Western Europe. The same word, mistletoe, refers to all of our native mistletoes. There are no introduced mistletoes. Mistletoes are native to Australia. Most of our species are endemic to Australia, and they've been here for a very long time. Most of the lineages you find here were well established well before Australia split off from Gondwana. So they've been here for quite some time. What I'm interested in and what most of my research group is focused on is this stuff. Not just the mistletoe itself, but interactions between mistletoe and various ecological partners, both in a pairwise fashion and a broader whole of system fashion. They're kind of unusual Australian plants in that they rely on birds, not just to disperse their fleshy fruits, but also as nectar feeders to pollinate them. Quite different interactions there. So they rely on specialists to disperse their seeds, mostly the mistletoe bird, but also a couple of honey eaters, whereas they rely on generalists as pollinators. So nothing just focuses on mistletoe flowers. It's the broad swathe of nectar feeding birds, honey eaters especially, that you see in woodland and forest canopies. They also snack on mistletoe nectar. They also pollinate those flowers. Bat, glider, various groups of insect pollinators may be involved, some species, but as a rule, they really are bird pollinated. So those are 2 interactions you might know of. There's many, many more. They have enriched litter. The way they draw moisture from their host is by retaining high levels of cations in all of their tissues that then pulls moisture from the host, trying to equalize concentration of cations down a concentration gradient. So they're enriched tissues. When those tissues are dropped, when those leaves, I should say, are dropped, they don't withdraw any of those nutrients. They don't care. They're parasites. So it's lots of litter, enriched litter, full of cations that they've taken from the host. They're returned then to the feeding zone of the host roots. But in that time, they're available to many detritivores. You see quite a different community of especially annual plants growing directly beneath the mistletoe than you see even a few meters away. So it's quite a different nutrient return rate there. Also from a structural point of view, these are semi-succulent plants with a very densely branched structure, quite different to the quintessential open eucalypt, acacia, casuarina hosts that tend to dominate Australia's woody flora. Many animals capitalise on that and use mistletoes as a nesting site, as a roosting site. About 3/4 of Australia's birds that are known to nest in foliage, in trees, nest in mistletoe. So it's a very popular resource as far as that goes. And then there's insects. There are a lot we don't know about insect mistletoe interactions. There are both generalists and specialists. Many insects, many butterflies will only lay their eggs in mistletoe clumps. They're developing larvae feed exclusively on mistletoe leaves. And in many areas, insects are probably one of the main controlling factors to stop mistletoes spreading far and wide. They eat a lot of them. And also possums, brush-tailed possums especially, love eating mistletoe. It's high in water, it's high in those cations I mentioned, and it's got none of the nasty allelochemicals and secondary compounds that eucalypts and many other Australian trees have. So it's good stuff, and possums go out of their way. They'll go through 4 or 5 trees just to get to one tree with a mistletoe in it. So that's a really quick snapshot of some of the interactions that relate to mistletoes. And there's broader effects of all these interactions. My group's looked at in some detail. We did a removal experiment, taking every mistletoe away from 20 woodlands and comparing it to a bunch of woodlands that we left untouched. Within 3 years of removing mistletoe at the woodland scale, those woodlands lost about 1/3 of their woodland birds. So really strong direct evidence of this role of mistletoe as an ecological keystone. But then we get to plane trees in Melbourne. So this project came about with Rodney Vandery and Lee Harrison at City of Melbourne, who were wanting to be proactive about some development that was going on in Melbourne. A lot of trees, mature established street trees, needed to be removed for upgrades to rail and road infrastructure. And they wanted to capitalise on what was going to be quite a disturbing effect and augment existing street trees, not just by planting other species that were going to be maybe a better fit as street trees, but also to use the existing trees that were there and improve their amenity for wildlife habitat. So nest boxes, chainsaw carved hollows inside those trees, but also mistletoes were sort of the 3-pronged approach of using the existing trees to make them better. And plane trees have an interesting backstory here. So Tim Flannery is quite opinionated on many things and has pointed out that really they might have the odd hollow, but they're effectively sterile. Nothing really eats them. They're a completely foreign plant. They're a nice street tree. They're resistant to pollution, all that stuff. But as far as wildlife is concerned, they may as well be made of concrete, was his point. So it was a nice opportunity to say, okay, well, can we take these, quote unquote, concrete trees and make them better for wildlife in our cities. So we sat down, looked at some maps. There's a map of downtown Melbourne. Melburnians will recognise some features there, and the pink is plane trees. There's a lot of plane trees in Melbourne. So we hunted around and found the best mistletoe to use for this exercise. There's a few mistletoes around Melbourne, maybe six species. This one is a fairly obscure one. Muellerina is the genus, Muellerina eucalyptoides. So this is a genus of Loranthaceous mistletoe endemic to southeastern Australia. And unlike the Amyemas, the Lysianas, some of the other diverse genera, they've got a ramifying Astelia that has multiple connections to the host. So they can effectively move through the host to track higher light conditions to prevent being shaded. And also they attach to the host mostly on the main trunk and the large branches. So not at the edge of the crown, so far less likely to cause premature branch fall, which for a street tree in a city with sports car parked underneath it is a problem the City of Melbourne really didn't want to have. So this attaches mostly to the interior of the tree and grows up from there. And then Lee Harrison led the charge with finding fruits, mature fruits within the City of Melbourne area. So this was really moved around. We weren't moving it to a new area. We were just moving it into trees that weren't hosting mistletoes for reasons that will become apparent. And we thought there might be a bit of backlash to this. We thought the wider community might say, sorry, you're putting what into the trees now? Have you lost your mind? So we built a nice website. We were ready for had some fact sheets. We were ready for some cranky citizens. And we got exactly the opposite. People are like, great, yep, cool. Can we have it in our place? What about in the parks? Other councils are like, yeah, we want to do that too. So we made some signs, look up and pucker up, capitalizing on the whole kissing Christmas business around some existing trees where there were established mistletoes. And there was a real groundswell of interest in this project. So it was covered quite well in the print media, and even Gardening Australia did a feature on this that really communicated this idea and the idea about mistletoes as native plants and as useful plants in our bushland to the wider community. So it was a very useful communication exercise in that sense. So in terms of the trial itself, we picked 26 plane trees in a variety of locations, all mature established trees with pretty open, well-lit canopies. And in July 2017, we planted a bunch of seeds on them. So 4 seeds per little inoculation site on 8 branches per tree. So 32 seeds per tree, 26 trees, that's 832 seeds planted in July 2017. And this sort of thing hasn't been done before. People talk about mistletoes as useful things. There's a few people have talked about using it for restoration, but this was the first comprehensive attempt to use mistletoe in a restoration sense. And it worked. It was a bit unclear how quickly it was going to work. Muellerine are very slow. They don't do anything very, very quickly. So that's after a year. We got 24 little babies, 24 seedlings on 15 of the 26 trees. And interesting, of those 15 trees that were successful, 12 of them had possum collars, those tubes on the main trunk to stop, too slippery to allow a possum to climb up. So we thought possums were pretty important factors in controlling mistletoes. We wondered if, and that's generally, and we wondered if possums were going to be one of the reasons why mistletoes were basically absent from downtown Melbourne trees. And there's some pretty strong evidence that it may well be possums that are involved. So of the 15 trees where mistletoe seedlings emerged successfully, 12 of them had possum collars. Field work went on just earlier this week, Monday and Tuesday of this week. I was back in sunny Melbourne to check on the babies. And what did we find? So this is one of few scientific studies that I actually live tweeted the findings. It's like, yes, found the mistletoe, and it's big. It was easily seen, and there were more. Oh my gosh, so much excitement. And then look, yes, all the mistletoes in the trees. So of those 15, hello, yes, I've got a fan here. Of those trees that hosted mistletoe originally, with seedlings, 5 of them, the mistletoes have grown to maturity, and critically, at least 2 of the plants that I could see with binoculars had mature fruit. So these are plants that established. These are plants that grew. They were successfully pollinated, and there's now mature fruit, hopefully seeding the next cohort of mistletoes in downtown Melbourne. So that's, in my money, that's a success. Just quickly, where are we going with this? While this work's been going on, a lot of other studies have been conducted on mistletoes. This one was led by Ross Crates as part of broader region honey eater recovery work, looking across 2,000 sites right across New South Wales and Victoria at areas looking at what nectar is available and what nectar feeding birds are feeding on. And we found that mistletoe was a really important source of nectar for many of these species. Areas with more mistletoe supported greater numbers of nectar feeding birds. And this really matters because the phenology of mistletoe is such that during droughts, other species simply don't flower, but mistletoe does. And parallel work that I've done with Francisco Fonteribel in Chile has shown that areas of mistletoes support more resident communities of nectar feeders that then go on to pollinate all sorts of other plants. And if mistletoe isn't there, those nectar feeders become more transient, and pollination success at the community scale falls off a precipice. So there's some interesting effects here on the wider community. And if we're concerned about our trees and our successional planning of our canopy trees, pollinators really need to be part of that thinking. But troublingly, as the study wore on, we found that during the drought, most of the mistletoes died. And we're not sure if this is just water stress or probably a combination of water stress and acute heat stress during heat waves. So there's the paper there, and that's something that we're going to be tracking through time with the Melbourne mistletoes. Cities, as you'd be aware, are often in the wetter, more fertile parts of landscapes. And I think cities have a real role to play as climatic refugia for many different species. And I think by adding mistletoes to the canopies of our street trees, we're adding that extra resource for our nectar feeders, potentially making our populations of nectar feeders much more resilient to these shock events, to these extreme events, weather events that we're seeing with, unfortunately, greater frequency and severity. And then the final element I wanted to leave you with, it's not just about nectar, it's not just about resources or the plants themselves. Microclimate appears to be a really interesting thing that mistletoes can affect. These plants are always transpiring. Their stomata on their guard cells physically can't close. So a mistletoe is always weeping water vapor, and a tree with a mistletoe in it is measurably cooler, especially during very dry conditions. And so there's a photo there of a booble cow camped out under a giant mistletoe on a 43-degree day. Adding mistletoes to street trees might take that urban cooling effect that we know trees can mediate. It might take that up a few notches. And we're going to look with interest at how these mistletoes in plane trees act when it gets stinking hot in future summers. And just for those interested, yes, you can grow your own. I would encourage you to do so. There's some very simple tips to follow there. You want ripe fruit. It needs to be nice and yielding, like an avocado in the supermarket. And you can keep it for a while. Just keep that skin intact. The minute the seed is out of the fruit, germination is triggered. And wipe it on the underside of a stick in a well-lit environment. About pencil thick, pinky thick, no thicker. And you're looking at about, depending on the species, quite a low success rate. So if you want a few mistletoes, plant a whole lot of seeds. And possums, think about possums, think about enemies, plant it on a spot where possums are less likely to visit. Yes, so look, I'll leave it there. Take-home messages for those of you keen on a quick recap. It's a native plant. It's a good thing. Steady source of enriched litter full of cations, so really accelerated return rates of many quite limiting nutrients in our depauperate soils. Lots and lots of nectar, especially at times and seasons when very little else is flowering and providing nectar for nectar feeders. So huge importance for pollinators. And they're cool spots in warm and getting warmer canopies. So yay for mistletoes. And if there's questions, look forward to hearing them. And if you want to know more about them, I'm quite easy to find, and I'm always happy to talk about mistletoes. Thank you so much.
That was very fun. Thank you. I learned a lot. I can't believe you had a 30% loss in woodland birds as well. It's pretty dramatic.
Indeed.
Very underappreciated. I think we need like a whole session on Australia's underappreciated genera. Now, next up, we are going to be talking about the Goldilocks zone for Senecio bayerianus. This one's by Damien Cook from the Wetland Revival Trust. It's a recording because Damien's actually in Western New South Wales at the moment with limited to no reception, but you can add any questions and we will pass them on to him.
I'm Damien Cook from the Wetland Revival Trust, and I'm a restoration ecologist, and I'm talking today about the stiff groundsel, or Senecio bayerianus. And the title of my talk is Bringing a Goldilocks Plant Back from the Brink. Stiff groundsel, Senecio bayerianus, likes things to be just right. So, and you can see here, there's a photo of the flower of the stiff groundsel, and a plant that's been planted in a wetland as part of a restoration project with one of the traditional owners, Laura Kirby, who helped to plant it. And you can see the plants establishing quite well there. And yeah, so I'll launch straight into from here. So the key points to this presentation are that stiff groundsel was once a very widespread species occurring throughout the Murray-Darling Basin and a little bit to the south, all the way from up near Bourke in New South Wales down to Murray Bridge in South Australia and through Western Victoria to sort of north-central Victoria. There was a lot of habitat loss and modification, which caused a catastrophic decline in the species, and it's listed as endangered under the Commonwealth Environment Protection and Biodiversity Act. Stiff groundsel was actually believed to be extinct until it was rediscovered in 1991, so it was collected fairly widely by botanists back in the 19th century, but then people stopped coming across it and no one saw it for quite a long time until it was rediscovered by a botanist in 1991 near Crop in Victoria. And now we know of a total of 6 extant remnant populations, which are all in Victoria. The environment of remnant populations I did a study of, and we were trying to create a habitat profile for the species, which was used to identify sites where we could reintroduce it, because a lot of the remnant sites are in road reserves or tiny sites that don't have a very good conservation security. So the stiff groundsel has now been re-established at 5 sites. We've planted about 400 individual plants grown from seed and cuttings collected from all of the remnant populations. And although we haven't had any seedling recruitment, never seen seedling recruitment in the wild, it's a strongly rhizomatous species and plants have been expanding rapidly at some of the reintroduction sites. So here's a map of the distribution of Senecio bayerianus, and it's got historic records as well. So you can see right up here near Bourke in New South Wales, there's a record over here in Sydney. I think that's a bit of an outlier. I'm not sure if that's actually accurate. There's a few records up here in southern New South Wales. There's a real cluster here, and this is the stronghold of the species currently, sort of north-central Victoria. There is this population down here in Ballarat. It's quite a good population. But yeah, historically it was over near Casterton and in South Australia over here down near Murray Bridge. So yeah, as I said, it was quite widespread, but most of these are historic records, and all of the more recent records are really this cluster here in central Victoria. So this is an aerial photo of what it looks like at Woods Point on the Lower Murray River, and near Murray Bridge in South Australia. So stiff groundsel was collected by Ferdinand von Mueller in that area in 1851, and you can see that's been fairly intensively developed for agriculture. So there's unlikely to be any remnant populations in that area, and the species actually is no longer found anywhere in South Australia or New South Wales. And this is the kind of classic plains country where Senecio bayerianus occurs. You can see this is after the 2011 floods, and so you can see across the landscape all the Gilgai depressions and larger wetlands. But you can also see in this image the cropping lines that are evident throughout the landscape. So this country's been heavily disturbed, and there's very little soil that's actually escaped disturbance from the modern European agricultural practices. So obviously the Aboriginal people would have harvested resources through this landscape and practiced their own style of of resource management, but yeah, never really ploughed up extensive areas. And so you can see that European land use has pretty much disturbed most of that soil and probably disturbed the habitat of the stiff groundsel. So why is Senecio berrianus, or the stiff groundsel, a Goldilocks plant? Well, just like Goldilocks, it prefers everything to be just right. Like Goldilocks had to have the porridge just right, and the bed just right, and the chair just right. So Senecio berrianus likes its environmental conditions to be just right. So it grows in a fairly narrow range of environmental conditions, mostly determined by the hydrology, so the wetting and drying of the wetlands, and the depth and the duration of flooding, and the soil type is really important. So all the remnant populations, you know of, are on relatively fertile, heavy clays, and those kind of environments have been pretty heavily modified for European agriculture. And it grows on the floor of some shallow wetlands and on the fringes of deeper wetlands, but generally in areas that are only inundated by up to about half a metre of water, or 50 centimetres of water. So it thrives when it's inundated, and particularly if it's flooded for up to about 6 months. But longer term inundation will kill the species, and it's actually disappeared from some locations after the 2011 floods. It was just flooded for too long, for too deep, and the plants didn't survive. It can also survive prolonged drought, but again, it appears to be declining in areas where river regulation has reduced the flooding frequency. So it doesn't like it too dry, doesn't like it too wet. It likes to be in the right sort of soil type, in the right sort of hydrology. So and it's also on the plains, and it's in lowland areas. So its habitat was targeted for destruction early on by the way Europeans managed the landscape. It doesn't like river regulation. It doesn't like wetlands being drained. So all of those factors have really reduced its habitat. So doing the habitat profiling of the wild populations basically involved going around to all the remnants population, all the wild populations, looking at the vegetation, doing sort of quadrats and species lists, looking at the soil types, looking at the hydrology of the sites, and trying to build up a picture of what the habitat for the species was. So this is one of the sites, one of the biggest populations, the big remnant populations is at Carrag in sort of near Corrop in northern Victoria, and it's a sort of a mosaic of red gum swamps and plain grassy wetlands, and it's quite intact in that image there. You can see that wetland's just been filled and it's just drying down, so quite diverse and herb rich. The other site that's just really quite intact is this plains grassy wetland at Miners Rest near Ballarat, and most of the foliage you can see in that image is actually the grey foliage is stiff groundsel or Senecio berrianus. So quite an extensive population there, but mostly clonal. Like it's each plant is rhizomatous and can cover quite a large area. So there's probably less than 10 genetic individuals at that site, even though it covers a fairly big area. And that population, even when it's been wet, I've never really seen flowering much, and I've been there to try and collect seed, and it doesn't seem to produce much seed either. So so these sort of habitats, these shallow wetlands on fertile lowland plains where the Senecio berrianus grows, there's lots of threatened species that live in those sort of wetland types. So things like brolgas and Australian painted snipe also favour those kind of habitats. So also around Corrop, we've got some roadside remnants. This is a roadside drain, and the Senecio berrianus is actually growing on an artificial embankment in this wetland here. And these artificial sort of steep gradient embankments seem to actually be one of the, there's a few of the wild populations are growing in these sites. So they've obviously been modified in the past, but they've provided an area for the Senecio to be able to survive. Particularly, this is on quite a large floodplain, which floods quite extensively in wet years. It's incredibly dry in dry years, but when it is wet, it can stay wet for a long time. So I think having those steep bank gradients allows the Senecio to be sort of in a place where it can still be moist but not be totally inundated for long periods. So so these sides of embankments on spoon drains and a levee seems to be one of the places where it's survived. And again, this is the population up near Lake Boga on a private property. It's basically growing on the outer wall of a river levee on the Little Murray River, and where it's probably getting a bit of seepage. You can see all the common reed or the Phragmites there, and it's growing in a bit of a different environment there, pretty much almost like a monospecific stand of Phragmites with a bit of the Senecio growing through in amongst it. And this is one of the main reintroduction sites that was done before I got involved with the species, which is at Two Tree Swamp. And again, it was an artificial embankment with the plants. You can see all those wooden pegs have a Senecio planted next to them. So it's done actually quite well on that embankment, but the Senecio was also planted in the natural bed of the wetland next to that embankment, but all of those plants drowned during the 2011 floods. So the only surviving plants are on that embankment, which is slightly problematic, because you can see there's quite weedy on that area. There's Phalaris aquatica, which is a big perennial grass, which could potentially take over that area. So but that was a successful translocation site. There was about 3 or 4 sites where it had been planted in that sort of crop Wallenjoe swamp area, but it seems, well, when I've been back to look, I can't find any plants at the other sites, so that's the only one that was really successful from that period. So one of the major threats to the survival of the Senecio borealis is the lack of genetic diversity. So all the wild populations are very small, down to less than 20 individuals. The populations are scattered across a large landscape with no opportunity for natural genetic exchange between the populations. And the wild populations produce very little viable seed. And they're also obviously subject to additional threats, including vegetation clearing, cropping, and altered hydrology. So one of the remnant populations up near Murrabit was destroyed in the last 10 years. It's probably inadvertently destroyed by the landholder. It was on private land. This weed invasion on these kind of soil types can be quite a bad thing if the sites are disturbed. Overgrazing and climate change, obviously being a wetland plant, and a plant that most of the wetlands it grows in sort of need good winter rainfall to kind of fill up the cracks in the clay and to flood. So so if we get more summer rain and less winter rain, that's not going to be great for this species. So what do? To try and increase the survival chances of this species, we actually started doing a bit of a dating service. So there was some research done, and the conservation geneticists at the Royal Botanic Gardens recommended cross-pollinating all of the populations, all the wild populations, even though some of them are scattered, by more than a couple of 100 kilometres apart. They said that basically the species would be doomed if we tried to break it into provenances and protect individual provenances, that we needed to mix the gene pool back up. So we collected seeds and cuttings, mostly cuttings from the populations, because some of them don't produce much seed, but where we could, we got seed. And we went around in about 2014 and got all the genetically distinct populations, and we gave them to several nurseries. Particularly, it's been successful with Newstead natives, and we've had native bees we've observed visiting the flowers, and when the Senecio is flowering. It's quite a late flowering species, mostly flowers sort of late summer, autumn. And subsequently those plants produce lots of viable seed, more seed than we ever got from the wild population. So from those we've grown about 120 seedlings, and mostly done a lot of growing from cuttings, and planting them into secure sites that were identified by the habitat profiling. And most of the sites we planted now actually receive environmental water, so we can bolster the chances of the survival of the species. And we've been reintroducing it, and the sites where we've been planting it and watering, we're getting now lots of viable seed from those sites as well. And last time I spoke to Frances from Newstead Native, she's got a seed tray of it coming up quite nicely now, whereas we used to get dribs and drabs coming up. So here's the Senecio. It's got a very strong white rhizome, and can spread quite rapidly over quite an area. So there's the Brappa traditional owners who've been helping replant the species at McDonald Swamp with a 3-year-old plant that's been put in as part of an ecological restoration project. these are the ones that we're below wetlands. We use guards to protect them, and yeah, we need to get them established. But after a while, they seem to be not to be too phased by native herbivore grazing. There's another one at the McDonald Swamp. Yeah, you can see there the inundation drowns out the weeds and leaves a nice clean area for the Senecio to spread into. That's it to get Johnson Swamp, McDonald Swamp again, and Johnson Swamp. And yeah, the best site we've got is Wirrallo Wetlands. It's and we actually deliver water to 11 wetlands at this site, and we've planted the Senecio borealis in a number of these where we've got probably the biggest and most genetically diverse population is on private land at this restoration site. So I'd like to thank Liz James and Neville Walsh from the Royal Botanic Gardens for providing advice for this project. Also thanks to Francis Incotta from Newstead Natives and Rod Cunningham and Sarah Bournes of Cosmos Seed and Soil for propagating the plants for release back into the wild. And also thanks to the members of the Barappa Rappa Re-Veg team for assistance with planting and looking after the reintroduced populations. Yeah, thanks for listening. I've been Damien Cook from the Wetland Revival Trust, and we're actually, if you wanted to help the Senecio borealis, visit our website to see how you could help us purchase Wirrallo Wetlands and ensure the survival of the largest and most genetically diverse population of stiff groundsel that is left.
Thank you to Damien. Really interesting that the unintended impacts of disturbance and habitat for threatened species. We're going to jump to Catherine Horsfall's talk now while we're thinking, backing up on urban impacts. So Catherine's going to posit a few ideas about the future of urban environments for conservation translocations. Catherine, if you are ready to go, that would be great. Catherine's from the University of Melbourne, by the way, and currently working on a PhD in urban restoration, I believe.
So thanks for having us. And of course, I'd like to pay my respects to the traditional owners on which this work was conducted. And in wider Victoria. So I, like David, am working in the big smoke, and I'm lucky to have for my PhD research a real world problem to resolve. And so the first experiment is based in Royal Park. That's the one I'm going to be talking about today, which is, of course, the traditional lands of the Kulin Nation. And it was part of a ring of parks reserved early in European settlement. But as you can see from the surveyor plan here, prior to European settlement, it was grassy woodland, and the park contains some remnants of this vegetation. To this day, it is managed as a natural parkland with open spaces interspersed with trees, although the canopy isn't quite as dense as what you see there. Oops, beg your pardon. All right, and so the park's had a lot of uses, including the Second World War as a military encampment. This aerial photograph shows all the soldiers' huts on what is now known as the Grassland Circle. And after the war, these were bulldozed into the ground. Unfortunately, they contained asbestos, and so the park still has soil contamination to this day. And the park is managed, as I mentioned, as a sort of naturalistic landscape. It retains the vision of naturalistic woodlands of indigenous eucalypts around the perimeter, with clear sightlines along ridges and at the top of hills. And the 1984 Royal Park Master Plan enshrined the idea of having a native grassland restoration on the highest point in the park, which is the Grassland Circle, and also sought increased species richness in the park. And that's backed up by City of Melbourne's Biodiversity Plan, Nature in the City, which seeks increased understory vegetation in the municipality. So because of the asbestos, we can't use some of the tried and true grassland restoration techniques that have been pioneered by the likes of John Del Pratt and Paul Gibson Roy, where we come along and we remove the topsoil and the nutrient-enriched topsoil and the weed seeds and sow into that, creating restoration similar to the one on the bottom right. So work at Royal Park to date has had mixed results in restoring grassland species to that grassland circle. The only area in which it really worked was on an old gravel car park, indicating that, yeah, you're up against the classic duo of soil nutrient enrichment and weed competition on the rest of that grassland circle. So one approach we might take is to build up upon the existing site profile with a nutrient poor substrate, such as a subsoil, which is that clod pictured at the top, which is also happily a waste product of the urban development world, or sand. And sand is an idea that's been explored in very much a horticultural tradition in the UK meadow movement. And here practitioners like Nigel Dunnett and James Hitchmore use seed, and they really stress sort of species selection to get things that will please the punters, pretty flowering heights and shapes and forms. But importantly, they use sand as a low nutrient substrate to minimise weed competition, and they irrigate to push rapid germination in an urban setting. So my idea is that instead of using international plant pellets, we use our Indigenous species to create a greater amount of these in our cities. So the Royal Park experiment asked a couple of questions. It sought to understand if we could use sand to support species-rich meadows. And importantly, we wanted to quantify the kind of labour inputs for the first critical year of establishment, where we're seeking canopy closure and a more resilient plant community in terms of weed invasion. And so we were playing around with, well, what depth of substrate would minimise weed emergence and support the species that we sowed. So it was, the experiment took place on a linear site underneath established eucalypts in an area that had formerly been mulched. We sowed 27 species of Indigenous wildflowers and grasses. And we conducted a species selection process, very much using that horticultural tradition of trying to choose things that would have a long season of flowering and interest to urban residents that live nearby. We carved up the site into 54 plots, as you see there, and we played with two depths of sand, 80 millimetres and 10 millimetres, as well as sowing identical seed mixes into the site soils. All right, so what we found was that the 80 millimetre depth of sand was doing a better job, a significantly better job of reducing the amount of weed competition, and then importantly, labour and thus cost of doing this kind of work. And while we found that our sown species were a little bit slower to establish on the greater depth of sand, by December they'd evened out and we had a fairly uniform canopy, which was dominated by a number of species. But of the 27 species that we sowed, we got 25 of them, still waiting on 2. And 3 of those are threatened species. Not in vast numbers, but certainly there. And we move on from this work to my current baby, which is playing with the subsoils that I mentioned earlier. So in both these experiments, we have had establishment of some threatened plants, but certainly we're trying to evoke threatened plant communities. It's definitely not the real thing, and it's not translocating plants from other environments per se, but it's working with seed to create new frontiers potentially for translocation. So if we can set these plant communities up to be robust, the question could be asked is, are they suitable sites for translocating into in the future? I'll leave you with that question, and thanks to City of Melbourne who support my work, and to you all for listening.
Thank you, Catherine. I like it that you went a little bit outside your comfort zone for that one. I appreciate it. You know, and I think based on the current state of the environment report, we probably need every idea we possibly can to conserve species. Now, our speaker, Amos, isn't able to join us, but Amos was going to be speaking about the Right Plant, Right Way project, which is a First Nations-led restoration program based in Victoria. We're going to put the link to that in the chat. That was a report last year that was produced in public, and I think it has a really great example of what can be done possibly Australia-wide to be working better with First Nations Australians as part of choosing the best species for the best environment. So because of that, we are 10 minutes ahead of time. I was going to kind of open it up to any questions. If anybody wanted to pop anything else in for Catherine now, or any of the other panelists. If anybody is still online panelist wise and wants to answer anything directly, you can. I will leave it for a minute or so, and then I will wrap up, and everybody gets a 10-minute early mark on a Thursday afternoon. Yeah, so Joe and Christine are popping all the links there in the chat as well, which is excellent. I had a question quickly for you, Catherine. Do you think that we can be doing any of this with shrubland species as well? I know there's been some work doing like pruning projects to replicate browsing and things in urban areas. Have you been involved in that?
I'll come in as a creepy disembodied voice.
Yeah.
Yeah, no, that's not. So we have a little bit of meadow competition going on at the University of Melbourne, but that's the woody meadow work you're referring to.
Yeah.
Which again is trying to make native plants, the focus of very resilient urban plantings that can withstand some of the rigors of an urban environment. And yeah, so they're looking at plants that will resprout after fire, and then those that, again, with that ornamental focus, trying to get them along rail sidings and all sorts of really hostile sites, but introducing a level of beauty and biodiversity as a result.
And have you had any kickback about having threatened species involved in urban landscapes, or everyone's pretty supportive?
Yeah, no, everyone's been very supportive. I mean, it's not been a widely advertised thing, but certainly, as a result of our seed production, which we had to do to generate the seed for this and subsequent sowings, we've got a lot of seed, for instance, of Rudidosis. So we could stretch it a lot further. We could be planting tube stock, and for those species that don't necessarily establish from seed, we could look at introducing more. But yeah, nothing's been taken from remnant populations. This is all cultivated seed.
Yeah, perfect. All righty. So if you have questions that you would like answered, you can always email me directly and I can pass them on as well. My email is translocate@anpc.asn.au. Joe or Chris will pop it in the chat. I've learned a lot. I hope, like me, you have some level of positivity moving forward for all the things that we can do within our toolkit to help reduce the slide in biodiversity in Australia. As with last week, it's going to be popped up on the ANPC YouTube channel. Last Thursday's session is already there, and this one will be there shortly. Joe and Christine, thank you very much for running around in the background, and to our amazing presenters.
Everyone. There's actually, there was a question there, but it's gone.
Oh, go for it.
Oh, there was a question for Catherine, but
go. Yes.
Answered it.
Yeah, so Shannon was just asking what kind of sand we used. It's a sharp sand, so it's fine-grained angular particles, low nutrients, and I think possibly not ideal in the long term for this kind of work, because that's a mined resource, and that's why we're looking at subsoils, which would be a recycled urban waste product.
That's a good idea. Any other questions before I say goodbye? Nope. All right, so Joe and Christine will be sending out a feedback form to you. If you'd like any changes made to the format or propose any topics for future symposia, please let us know. ANPC website, you can find out about future events, also a e-newsletter. Also, you could become a supporter of ANPC because it's a not-for-profit and we work Australia-wide for plant conservation. Okay, so now it's probably time to say goodbye. My final thank you for joining us, and have a wonderful rest of the week. Thank you all.